speakers, our dear delegates, and all the students. We warmly welcome you to the chilly morning, and we are beginning with our session 13 of the day 3 of ICoM 26 conference. We are beginning our session, which is titled as Neuroscience, Brain Health, and Neuro-Oncology. This session brings together fundamental neuroscience, clinical perspective on brain health, and advances in neuro-oncology, highlighting how molecular and translational research is shaping the future of neurological care. We warmly welcome the chairperson and the co-chairperson of this session. It is our privilege to begin our session by introducing the chairperson of the session, Dr. B.N. Malik. Dr. Malik is a highly distinguished neuroscientist and currently serves as director and the professor at the Amity Institute of Neuropsychology and Neurosciences. He is internationally renowned for his pioneering contributions to the sleep neurobiology, with particular emphasis on the regulation and functional significance of rapid eye movement sleep. Before joining Amity, Professor Malik headed a leading sleep research laboratory at Jawaharlal Nehru University, New Delhi, where he established one of India's foremost research programs in sleep and brain physiology. Through decades of research, teaching, and leadership, he has played a major role in shaping sleep research in India and continues to be a guiding force in neuroscience education and innovation. We request Dr. Malik to please join on the dais.
And please put your hands together to welcome him. All the students are requested to kindly be seated in the front rows. All the students at the back are requested to kindly be seated in the front rows.
Please get up and come onto the front rows. We are requesting you to take the front seats.
So our chairperson is joined by co-chairperson, which is Dr. Gundeep Kaur from University of Texas, USA. Dr. Gundeep Kaur is a research scientist in the Department of Epigenetics and Molecular Carcinogenesis at the University of Texas MD Anderson Cancer Center, Houston. She's a recent awardee of the prestigious Ramanujam Faculty Fellowship. Her research investigates the molecular, biophysical, and structural principles that underlie chromatin remodeling, heterochromatin silencing, and epigenetic regulation in cancer and rare genetic disorders. By integrating advanced structural biology tools such as cryo-electron microscopy, X-ray crystallography, and SACS, along with quantitative biochemical approaches, Dr. Kaur uncovers how chromatin remodelers and chromatin binding complexes regulate genome accessibility and maintain epigenetic homeostasis. In addition to the Ramanujam Fellowship, Dr. Kaur has been recognized with several competitive honors, including the Newton Bhabha Fellowship at the University of Oxford, UK, DST Inspire Fellowship at CSIR IMTech Chandigarh, Teach for India Fellowship, and multiple institutional awards. Her work has yielded key structural insights into chromatin-associated complexes and has been published in leading journals such as Genes and Development, Nucleic Acid Research, Science, PNAS, Science Advances, eLife, and JBC. Beyond research, she actively contributes to scientific leadership as a reviewer, editorial board member, conference organizer, and mentor. Her long-term vision is to advance mechanistic understanding of epigenetic regulation to enable the development of novel chromatin targeting therapeutics. With this glorious bio sketch, may I request Dr. Gundeep to please join Dr. Malik on dais. So the session is now officially handed to you both. Thank you.
Hello, good morning, everybody. As you have heard, I am B.N. Malik, and here is Gundeep along with me. In this session, as it has been already introduced, we are talking about some neuroscience and brain health. There are 5 speakers. 2 of them, I'm told, will be online. The first one and possibly the third one. So we start with the session with your permission. Okay, thank you very much. Now, Dr. Gundeep will be introducing the first speaker.
Hi, good morning, everyone. It's my privilege to introduce Dr. Akhil Varshney. He's an assistant professor at the Center for Advanced Vision Science, Department of Ophthalmology, University of Virginia, US. He has more than 12 years of extensive post-PhD research experience in molecular biology and ophthalmology. Okay, sorry about, excuse me. So his research mainly focuses on retinal diseases such as age-related macular degeneration and retinoblastoma. He studied RNA biology during his PhD at JNU, New Delhi. During his postdoctoral training in ophthalmology research, he investigated the underlying mechanisms of retinal degeneration disease at both the University of Kentucky and University of Virginia, US. As a scientist and a PI at Dr. Shroff's Charity Eye Hospital in Delhi, he helped establish a fully functional research lab from scratch and conducted extensive research on various ophthalmic diseases of the cornea and retina. Throughout his career, he has been awarded several prestigious grants from the Government of India, including the UGC Meritorious Fellowship, TBT RA Fellowship from Postdoctoral Research in India, Ramanujan Fellowship as an early career PI, and a research grant from ICMR as a principal investigator. These grants have provided invaluable support for pioneering research in his field. He has contributed to the publication of 28 research articles in high-impact journals, accumulating over more than 1,200 citations and achieving an age index of 17. I would like to welcome Dr. Akhil Varshney. So please, it's open for you here. Sorry to interrupt. So you will be given 20 minutes, and after 20 minutes, there will be a little buzzer indicating that you have only 3 minutes left to wind up.
Yeah, sure. So thank you very much for the introduction. So I hope my slides are visible. And am I audible?
Yes, yes.
Yeah. So good morning, everyone, and respected chairpersons and audience. Thank you, organizing committee, for providing me this opportunity to present my work on retinoblastoma. And it's 11 p.m. in the night here, local time in US, and I will try my best to inform you about our work on retinoblastoma, and it's clinically relevant translational work where we try to develop CSF-based liquid biopsy for the prognostication of retinoblastoma. So, retinoblastoma is the rare, but it's a horrible pediatric cancer. Its rate is about 1 in 17,000 live births. And generally, the disease occurs at very early childhood, like age 0 to age 5. And if it's undetected and if it's advanced to a later stage, it's a lethal and it causes mortality at very young age. So generally, patient present with the leukocoria, that is the white pupil. As you can see in this image, the white pupil rather than red reflex when you capture the image of a person. This white reflex in the pupil of the eye of the small baby indicates the presence of retinoblastoma inside the eye. The below is the image of the fundus image, where you can see the background retina, and the white is the retinoblastoma tumor. This is the another image of retinoblastoma, where the background pink is fundus and the white is the retinoblastoma tumor. This is the third image, is the patient underwent chemotherapy and the retinoblastoma tumor regressed. So retinoblastoma occurs because of the mutation in the RB1 gene. As everybody knows that RB1 is the famous tumor suppressor gene, and like other tumor suppressor gene like P53, P10, those are involved in many other cancer type. But there is a unique difference between this RB1 tumor suppressor gene and the other tumor suppressor gene like P53 and P10. If you can see, the mutations spread throughout the gene of the RB1 from the exon 1 to exon 27. So the first peculiar feature of mutation in the RB1 is that there is no hotspot. It's generally spread throughout the gene. And the second peculiar feature of RB1 is 90% is the stop codon. So if you see this pie chart, 54% nonsense and 36% frame shift. Both of these contribute to the stop codon mutation. And only remaining 10% is the missense mutation. So because of the absence of full-length functional RB1 protein in those patients caused development of RB1 tumor at the very early age of life. It causes the loss of the eye or the life if not treated on time. So retinoblastoma, as I said, it can occur in the eye. So it can occur in single eye, then it's called unilateral. And if it occur in both eye, then it's called bilateral. So in this pie chart, you can see the 60% cases are the bilateral cases, only 40% are the unilateral. That means bilateral, there is a high chance of retinoblastoma in both eye compared to single eye. Second thing, the retinoblastoma is also a genetic cancer, that hereditary cancer. So it's caused because of the genetic mutation in the RB1. And there are sporadic cancer, that is the somatic RB1 mutation. That means those mutations are not present in the parents. So again, here you can see in this pie chart, 64% are the germline and 36% are the somatic patient. So germline patient, that means hereditary retinoblastoma, always lead to the bilateral retinoblastoma. As well as you can see here in the orange color, the germline bilateral. And all the unilateral cases are because of the somatic mutation. So somatic mutation leads to the unilateral, that means single eye retinoblastoma, and the germline RB1 mutation, that means the mutation coming from the patient leads to the both eyes retinoblastoma. So why it is important to know this? Because the life-threatening bilateral is considered less life-threatening compared to unilateral, because when the parents has a history of retinoblastoma, then the child can go routine examination, and in bilateral cases, generally they detect the tumor at very early age and can be treated successfully, and the life can be salvaged. But in unilateral cases where you don't know, because the parents are totally fine, but somatic mutation happened only in the children. So generally in unilateral cases, patient present at the later stage with the advanced tumor, and that's where the there is the life threatening at unfortunately cause the patient death. So let's talk about the retinoblastoma Indian scenario. So around 1,500 new patients of retinoblastoma are diagnosed in India every year as compared to 300 in United States. So generally this retinoblastoma disease is considered as a disease of the low and middle income countries. Maximum of the cases reported from the African and Asian countries, and very few, the less abundance in the Western countries. So in India, retinoblastoma is more commonly present with extraocular and metastatic disease and have a poor outcome with 50% mortality, and compare to Western counterpart where retinoblastoma commonly present with intraocular and survival rate is more than 95%. So definitely in Indian scenario, need to do more research on the retinoblastoma because that disease is more prevalent and causing death in Indian scenario. So in India, because since retinoblastoma present at the advanced stage, and in this figure, you can see the eye is connected to the brain by the optic nerve. So if the tumor is already advanced, then tumor cells migrate from the optic nerve to the brain and cause the central nervous system metastasis, and that cause the patient's death. So there are 9 to 45% of cases in the that is intraocular extraocular extension at presentation, and 30 to 71% of mortality at stage 3, and around 100% if patient present at stage 4, and again around 100% if optic nerve found positive with the cancer. And almost all around uh, India, the several cases of retinoblastoma reported. So overall survival outcome stage 3 is 30 to 71%, and stage 4 only 0 to 8%. So there is unmet need to develop strategies for management of these advanced cases of retinoblastoma that cause the high mortality rate. So when retinoblastoma at stage 3, stage 4, that cause the CNS mets, and most of the diagnosis by the imaging modalities like MRI. So in this figure, you can see the T1 weighted image that's showing the left eye intraocular mass. As you can see here, if you look at this eye versus this eye, you can see this is complete black. That is the normal eye. But here in this eye, if you look, this whitish thing, so this is the intraocular mass with the retinal detachment, and that is the example of exophytic retinoblastoma. Exophytic retinoblastoma means so retinoblastoma is two type, endophytic and exophytic. Exophytic is something that is growing over the retina. And then you can see this is the optic nerve, and here by this image you can see the optic nerve sheath involvement and swelling of the optic nerve that can lead to the intraparenchymal and leptomeningeal disease in the if CNS mets happened. So extraocular extension of tumor with optic nerve involvement, that's considered as a high risk of CNS relapse. So it's very important, if we want early treatment, we need to detect of minimal disseminated disease. How we can detect? So current conventional way is the CSF cytology. So CSF is collected from the patient and then analyzed for the atypical cell. But unfortunately, in many cases, this conventional CSF cytology not able to detect the minimally disseminated CNS disease and leads to the death of the patient. So minimal CNS dissemination without radiological evidence can go undetected and the poor outcome. So this is an example of the patient presented with advanced tumor, and MRI again with the right eye involvement of the retinoblastoma, and MRI showing proximal optic nerve involvement with no CNS involvement, and CSF cytology was negative. This patient underwent chemotherapy. It responded to chemotherapy, and you can see the thysical eye after chemotherapy. But unfortunately, after 4 months, the tumor reoccurrence happens. After surgery, patient come back with local reoccurrence, and after a few months, patient died due to the CNS mets. So now the question is that if there was a test that could detect CNS involvement at early stage, then one could prognosticate better and probably treat the metastatic disease at early stage. Treatment at early stage could provide different outcome. So if we able, since both MRI and the CSF cytology was negative, so we didn't know that this patient can go tumor recurrence. So if a test can tell about whether that patient will go in tumor recurrence, then the aggressive treatment can be performed to save the life. So the question is, can liquid biopsy predict the risk of metastasis or predict preclinical mets? So what is liquid biopsy? Liquid biopsy is the procedure to detect the cell-free circulating tumor DNA from the bodily fluid like blood, CSF, or urine. So that body fluid contain the cell-free DNA from the normal cells as well as the cell-free circulating tumor DNA from the cancer cells. And by the sequencing method, we can differentiate between the cancer cell derived circulated tumor DNA from the cell-free DNA that coming from the normal cells. So we developed this methodology to investigate this problem. So we collected the three compartment fluid and the tissue. So we collected the CSF as a part of metastatic workup and the blood samples and the tumor from the enucleated eye. Those patients underwent enucleation because of the advanced retinoblastoma. Then isolated the cell-free DNA from CSF and cell-free DNA from the plasma and do the NGS sequencing and the RB1 mutation identification. At the same time, isolated the tumor DNA from the tumor for the validation by the Sanger sequencing. We used a robust pipeline and methodology for the detection of cfDNA and variant identification from the cell-free DNA isolation from the well-established Kaizen kit and the library preparation by the Agilent SureSelect XT target enrichment kit and variant identification by the robust bioinformatic pipeline and mutation concordance check by the CSF, tumor, and the blood plasma, whether the same mutation is present from the CSF and the blood and the tumor can tell that the mutation detected in CSF is not because of the error in the sequencing, but it's a real mutation, that tumor DNA coming from the tumor itself. So the purpose of the study was to evaluate the usefulness of CSF ctDNA liquid biopsy as a modality for detection of early CNS dissemination and prognostication of retinoblastoma patient. So this is the patient that presented the first time with the left eye involvement. As you can see this MRI image with the white color, this is the left eye that is involved with the retinoblastoma. So blood samples collected at the time of presentation and the sequencing was done and RB1 mutation was identified. This patient underwent chemotherapy and respond to chemotherapy, but this patient come back again with the reoccurrence and the plasma and the tumor sample was collected at the time of reoccurrence as well as the CSF. And after sequencing, we find the same mutation that present in the plasma, CSF, tumor. So this means this DNA is coming, mutated DNA in the CSF is coming from the tumor. So unfortunately, the conventional metastatic workup, including MRI scan, CSF satellite did not detect any metastasis, but later on patient died due to the disease, recurrence and CNS mets. Despite CSF cytology negative, we identify RB1 mutated DNA in CSF liquid biopsy, and of course, germline RB1 negative. So all those patients we studied are germline RB1 negative. So this is another example of the patient with the 7-year-old female. Again, the same case of metastatic workup, including MRI and CSF cytology, was negative. Patient developed CNS symptoms and succumbed to the disease. Germline RB1 negative, and we found the RB1 mutated DNA in CSF and the tumor as well as blood. So similarly, we did perform this analysis in 10 patients. So we take the 10 unilateral cases, as I introduced in the initial slides, that unilateral means one eye, and sporadic somatic mutation. So that means these are the patients, those are on the high risk. So 10 unilateral means the retinoblastoma in one eye, and the RB1 mutation, somatic mutation, means mutation was not present in the parents. So these patients generally present in the clinic at the advanced stage. So we chose those 10 patients, non-germline stage 3 RV negative, and all of those 10 was the negative for the CSF cytology. The average age is 4.5 year, 2 to 8 year. Follow-up duration 4 to 21 month. Average CFDNA isolated from CSF is 2.6 nanogram, and the from the plasma is 14.2 nanogram. So out of 10 stage 3 retinoblastoma patient, we found 5 patients with the CSF ctDNA positive and 5 patients with the CSF ctDNA negative. So out of those 5 patients, those are positive for the ctDNA. 5 patients died, and so all 5 patients died because of the disease, and those 5 patients where ctDNA was negative, only 1 patient died and 4 patients survived. So on that basis, we calculate the sensitivity and specificity of our assay. Sensitivity is 83% and specificity is 100%. So the and the variant allele frequency VAF, we found range from 6.1 to 61% in the CSF, and VAF in plasma from 4% to 37%. So this study give the confidence that CSF liquid biopsy can be used for the prognostication of advanced retinoblastoma. Further, we want to see the concordance of CSF ctDNA from plasma and the tumor RB1. So as we know that in every case, all three, the CSF, blood, and the tumor tissue is not available. So out of 10 patients, 7 patients we have the CSF and blood, and 3 patients we have the CSF and the tumor tissue. So those 7 patients with the CSF and the blood, 3 was the ctDNA positive, and we found out of these 3, 2 patients with the same mutation in the blood and CSF. In the 1 patient, the mutation present in the CSF but absent in the blood, because maybe that the dissemination of the tumor can happen only in the CNS, not in the blood systemically. While those CSF and the tumor DNA, we found CSF positive too, and that both CSF positive is same mutation is present from the tumor DNA derived from tumor. So this is the high level of concordance, that 66% with concordance between the CSF and the blood, and 100% concordance between the CSF and the tumor. So although the size is small, so next we are expanding the study for the large cohort to validate as a routine practice in the clinical management of this patient. So overall, ctDNA detection to symptom onset is 7 weeks and range 3 to 40 weeks, and symptoms onset to death is 2.6 weeks and range from 1 to 4 weeks. So CSF-based liquid biopsy could detect minimally disseminated CNS disease by mean of 7 weeks prior to symptom onset or detection of CNS dissemination on neuroimaging. So the conclusion is the CSF liquid biopsy based on RB1 mutation in ctDNA detected minimally disseminated CNS disease with 83% sensitivity and detection of minimally disseminated CNS disease, and it has a role in early treatment. So by this liquid biopsy, we can able to detect the CNS disease early and able to salvage the life of those kids. And further validation and correlation with imaging and the clinical finding is required. So I would like to acknowledge and thanks Dr. Shroff Charity Eye Hospital. Dr. Seema Das is the surgeon and the director of the oculoplasty ocular oncology. And Dr. Ravinder is director of histopathology, Govind G.V. Pant Hospital. Dr. Arpan Gandhi is the director of histopathology at Dr. Shroff Charity Eye Hospital and other student. And the funding from USAID, ICMR and DST. So thank you.
Okay, thank you, Dr. Varshne. Now, I believe you would like to answer a few questions if the audience has, but since we have exceeded, I think we can take 2 questions. Yes, please.
Hello. Hello. Hi, Akhil. Very glad to have you again in our international conference. This was a very nice study that you linked retinoblastoma with cerebrospinal fluid tumor. But my one query is that is this presence of tumor cells in the CFF affecting the brain function? You reported the death of the some patients, but is it clinically affecting the brain function? This is one question. And the retinoblastoma treatment is more or less you have to take care of the mutation, but chemotherapy but is some adjunct therapies will be useful to cure and add to the treatment of the disease? Okay.
Yeah, sure. So let's first I will answer your first question that how the tumor the metastasis into the brain caused the death of the patient, because it's like a brain tumor, right? So when the cell when the cancer cell migrate from the eye to the brain via the optic nerve, it causes the brain tumor. And that brain tumor is not because of the blood-brain barrier and all other thing. The drug generally not very effective for the brain tumors, and that's caused the death of the patient. So that's the answer of the first question. Second question is that I'm sorry, I forgot. What was the second question?
The adjunct therapies.
Adjunct
Will the
therapy.
adjunct therapies?
Yeah. So current standard of line for therapy for retinoblastoma is vincristine, etoposide, and carboplatin. That is the current line of therapy, chemotherapy. Generally, patient from the stage 1 and stage 2 intraocular retinoblastoma respond to this therapy, and generally tumor regress after chemotherapy. But this chemotherapy, as I shown in the patient of the advanced retinoblastoma, can initially it cause regression of the tumor, but afterward the tumor reoccur and cause the death. So the adjunct therapy, people use the radiotherapy by the plaque, and the localized therapy by the intra-arterial chemotherapy. The artery is going to the eye directly. So by those methods, generally clinician treat the retinoblastoma patients. Yes.
Thank you. Thank you for your
Yes, the
presentation.
second question here.
Good morning, Dr. Varshne. So I have two questions, short questions. So first is there any correlation between the number of mutations which are present in the gene and the bilateral and unilateral occurrence of the carcinoma tumor? And second is, what are the PPV, the positive predictive value of your test?
Yeah, so in the first question, there is the unilateral and bilateral disease is because of whether it's the mutation is germline or somatic. It's not because of the number of mutation. It's independent. So generally, people have detected on a single RB1 mutation is sufficient to cause the retinoblastoma. If biallelic, if the RB1 mutation occur in both the alleles, its total function loss of the RB1 can result into the retinoblastoma. Now, the unilateral and the bilateral retinoblastoma can be depending on whether it's a hereditary, genetic, that is a mutation coming from the parent, it's bilateral. And if it's somatic, the mutation happened only in the child, then it's a unilateral. And so what we calculated, the second question is, what we calculated is the sensitivity and the effective. So what we found is sensitivity and specificity. We found the 83% sensitivity and the 100% specificity.
Okay, I think, thank you, Dr. Varshni, to keep you awake. And it looks you are quite awake now and you are not tired, but I think you can, there may be more questions, including from me, but then we have to keep going. Thank you, Dr. Varshni. We will move on to the second speaker.
Yeah. Thank you.
Thank you. Yeah.
So the next speaker for today's session is Dr. Biswaru Basu. He's a senior scientific officer at Department of Neuroendocrinology, Chatranjan National Cancer Institute, Kolkata. He did his bachelor's and master's in zoology from University of Calcutta and PhD from Jadavpur University with CSIR Net Fellowship. He has 15 years of post-PhD academic experience, including postdoctoral training at NIH US and independent teaching research at Amity University, Noida, and Chitranjan National Cancer Institute, Kolkata, where he joined in 2018. He's serving presently as the Senior Scientific Officer. He's also leading the Department of Neuroendocrinology and Experimental Hematology at CNCI. His group includes 4 PhD students and 1 research scientist. His research interests mainly focus on autophagy regulations in breast tumorigenesis, neuroendocrine immune crosstalks in ovarian and gastric cancer progression, neurocognitive impairments due to glioma and chemoradiation, mitigation of chemotherapy-associated neurotoxicities and bone loss, and understanding abnormal tissue regeneration in traumas, including burns and radiation. He has received number of grants from prestigious agencies like DST, ICMR, DBT, SERB. He's also a recipient of DST Young Scientist Scheme, India Bioscience Regional Young Investigator Meeting Grant, and DHR HRD Short-Term Visiting Fellowship. He has published more than 30 papers and is a reviewer of many prestigious journals. He's going to talk about neuroendocrine modulation of dopamine and insulin in wound tissues as an effective homeostatic strategy with tissue regenerative potential. Over to you, Dr. Basu.
सर उस पे पॉइंटर नहीं चलेगा। हाँ, मतलब लाइट नहीं चलेगी, पॉइंटर इसमें दे देता हूँ।
Hello, I'm audible? Yeah. Okay. So good morning, a very good morning, and thanks to the organizers for inviting me for this beautiful conference. And before that, before I go to my speech, I like to say, like, back to Amity is like a homecoming, and it's like because particularly the Amity Institute of Biotechnology, where I have been for at least 3.5 years. So it's always a pleasure to come back here. Today, I'm going to talk about neuroendocrine regulations in the wound healing scenario. Because I am a cancer biologist, but our group also works in some of the neurobiological as well as tissue engineering aspects. So today's my talk is on tissue engineering. Yeah. So basic idea of wound healing is like there are different phases that can be seen here. I'll go very fast in this introduction part. These are the tissue homeostasis, followed by inflammation, migration of the inflammatory cells, proliferation and angiogenesis in the tissue beds, and like this is like the major part, and then followed by the tissue, like these are the fibroblasts or the keratinocytes, which help in the remodeling of the tissues. And these are actually intricately balanced in different time frames that can be seen here. That is a generalized idea. But there are many conditions like in diabetes or some other neurological conditions or maybe burn trauma where there's a dysregulated version of this can be happen, where you can see like different cells and different cytokines and other players which are actually regulating this scenario. These are dysregulated by both in the time frame as well as in different type of the functionalities also impaired. So there may be tissue scarring or improper tumor modeling, or there may be chronic wound, which is like wound which is not healing. There's a chronic inflammation going on. So ultimately there is inflammation and angiogenesis. These are two important events required for proper tissue regeneration. So what are the neuroendocrine regulation? I'll go try to understand. So neuroendocrine regulation is an emerging area in tissue homeostasis, where we can find out that the cutaneous, the skin is actually innervated with nerves. Nerves actually together with endocrine system, and also other different body system, they act as a homeostatic entity. So it's a like an endocrine and as well as there's this nervous system. They act together to actually, this is a very emerging area of any type of the skin organization or skin functionality. So there are multiple reports that has emerged that there is also like a brain function, which is also acts with the peripheral wound healing. So there is directly involves these stress signals, this hypothalamic gonad axis, and as well as this the global scenario of the wound healing is regulating also with the homeostasis of the local wound healing possibility. So it has been found that wound healing is actually delayed in denerved skin. That highlights the significance of the cutaneous innervations. There are also different players, like different cytokines, different neuropeptides, which are actually involved in the skin maintenance of the homeostasis. And when there is a dysregulation, this can actually be found in the effectively modulation. So there are neuromediators like these, VDNFs, CRH, NK. There are a long list which has been reported in healing. There are endocrine factors also, like different factors like we have already discussed, like the growth factors, which are secreted by different type of cells. These are also important players in the wound healing. So I'll go also very little bit on the physiology of burn wound healing, which is like one of the main part of my discussion today. So cutaneous wound healing, like we have said, the burn wound healing also is a major problem in all of the different parts of the world. So there are epidermal, dermal layer are involved in the third degree of the burn. So that is a reepitheliation is extremely slow, and that costs a huge amount of the expenditure for the hospitalization, and there is patient mortality. And there has been many solutions, particularly the silver X. The silver has been used for this wound healing. But there needs many different type of innovative solutions also. So what happens? The neuroendocrine changes after there is a burn injury. So you can see like there are immune and inflammatory responses that are surged, and there is post-burn hypermetabolic changes. Like hypermetabolic, the body comes into a catabolic state, which we can show here. I mean, you can see here, as we have mentioned, CNS, intestine, muscle, liver, kidney. It's a kind of like when the whole body is involved, burn is involved. And we can see like here, the serum catecholamine, glucocorticoid, cytokine, insulin, these are upregulated. And there is a hyperglycemia and organ dysfunction that actually leads to the sepsis and the death. That's the main problem for the burn, because before the burn can take time to heal, the systemic problems that actually led the patient into the mortality. So there are many different type of hormones, and that has been listed here, which actually are responsible for this burn injury. Particularly the catecholamines that are searched for different long time, insulin, which is also searched for quite for a long time if there's a burn injury. These are the two important factors which actually I wanted to discuss, and whether modulation of these two factors can lead to some of the solution for wound healing. First is like the dopamine, that is like what our interest that I will be like discussing in cutaneous wound healing. What is dopamine? Dopamine, as we all know, dopamine is a neurotransmitter, catecholamine neurotransmitter, which has both the central and the peripheral dopamine are there with the respective functions. Central dopamine, we know that is a mood, motivation, motor reflex. These are the particular areas which controlled by the dopamine. Peripheral dopamine also has some significant role, particularly immune functions, different types of the blood pressure, renal function that has been found. What I want to also discuss here, dopamine acts through its receptors, D1 class of receptors and D2 class of receptors. D1 is actually activated by adenyl cyclase, cyclic AMP pathway, activation of the PKA pathway, and D2 receptor class, they actually act on downregulation of the adenyl cyclase and cyclic AMP pathway. But together with that I have highlighted here, there is another like function of the D2 dopamine receptors, which has been elucidated in many of the functions and as well as in our lab also. That is, D2 dopamine receptors can downregulate the activated VEGF receptor phosphorylation, and that can actually, which is also responsible for the angiogenesis in different type of tissues, and which I'll be going to discuss a little further. So our lab actually in my PhD lab that was involved in understanding the role of dopamine, peripheral dopamine in tumor angiogenesis. So it has been found that the catecholamines neurotransmitter dopamine through its receptor dopamine D2 receptors can actually downregulate. The function has been, it has been given here that dopamine receptors are and dopamine as well as dopamine receptor expression is downregulated when the cancer progresses, like particularly the stomach cancers, and even that has been found in ablation of the dopaminergic nerves that can also stimulate the tumor growth. And if we externally administer the dopamine D2 receptor agonist with activation of this, that can restore the that can counteract the tumor growth. By what mechanism? That counteracts the VEGF receptor phosphorylation as well as the MAP kinase and focalization kinase phosphorylation, so that actually hinders the endothelial cell proliferation and endothelial cell migration. So this was the hypothesis that, this is a working principle that we already we have in our lab. So we wanted to understand, in case of a wound healing, there is a need of angiogenesis. And we are showing in the tumor condition that we want to prevent angiogenesis. So what happens if we can actually do the reverse thing, like understanding if we can administer the dopamine D2 receptor antagonist, which whether it can accelerate the, it can actually propagate the angiogenesis more. So these are the initial experiments, quite old experiments, where he showed that dopamine D2 receptor antagonist can actually regulate the wound tissue. This can actually regulate the angiogenesis, and the wound is actually healing faster. So I'll go very fast. This has been found to regulate this HOXD3 and alpha-5 beta-1 expression, both in wound bed and HUVEC cells. This has been published earlier. Also, the dopamine D2 receptor can regulate the mesenchymal stem cell mobilization in the wound angiogenesis. So this is the working model. And then we also wanted to understand what is the role of insulin in the cutaneous wound healing. When there is a so here we wanted to understand both in the diabetic and non-diabetic condition. So in diabetes, there is a prolonged inflammation, there is impaired proliferation that has been found. So there is a continuous angiogenic, this pro-inflammatory condition is found in diabetes. But side by side, there are many reports are coming like insulin has a anti-inflammatory response, anti-inflammatory role, as well as helping in the cell proliferation and survival. So we wanted to understand like topical insulin. whether not the systemic, the topical insulin, whether it has an anti-inflammatory, anti-inflammatory role, which can we can help or modulate into the wound healing scenario. So this is like the insulin. So we made some nanoformulation, silver nanoformulation of the insulin, which actually can be tested with different type of the dose and different type of the concentration and different days. And we found out that this insulin nanoformulation, this is the ICNP, as well as the insulin, both in the HECA cells, these are skin cells, it can show that faster wound healing activity, and it is biocompatible. So this principle we also transferred into the diabetic and non-diabetic animal model, where you can find that this insulin as well as insulin nanoformulation treatment could accelerate the wound healing in diabetic as well as the non-diabetic model. And that was actually correlated with the inflammatory cell migration at the in the wound bed. And also simultaneously, we could find out that pro-inflammatory cytokines level, IL-6, TNF-alpha, that were downregulated, and the anti-inflammatory cytokine, that is IL-10, has been upregulated in the different time points. And that is done by the insulin. So topical insulin can regulate the this inflammation, the prolonged inflammation that is found in the in case of a wound scenario, as well as the diabetic. So next, we wanted to understand if the exogenous administration of this insulin or the dopamine antagonist, eticopride, that has been used there in intraperitoneally, whether that can modulate the burn wound remodeling, and what is the molecular insight, and whether it's a critical type of administration, and whether we can translate the knowledge or extrapolate the knowledge with a human scenario, because we cannot intervene in the human. What we can do, at least, we wanted to understand. So two of my students, Sandeep and Vijana, they worked in this particular question. Here we are showing that insulin and its nanoformulation showed accelerated burn wound healing. In a burn condition, burn was a third-degree burn, was inducted in a, that's a very known, this is a standardized model in a mice model. So this is the histology of the unburned control. And this is the burn. We can clearly shows that the epidermal and the dermal layer has been abolished. And this is a burn condition. Here we have used the, compared to the control, we have used this empty insulin the formulation, insulin nanoformulation, and positive control as a silver X, which is a silver ointment that is used for the, particularly for used in burn conditions. We can see that this insulin nanoformulation as well as insulin shows a higher amount of in the burn condition, higher amount of burn wound healing. This is evident from this graph also. We also further taken what is happening in different days and conditions. We have actually evaluated all the different type of days, histologically, as well as their understanding. So we found that at day 10 for this particular and day 20 have significant role in acceleration of this burn wound healing, which has been found in the histological condition, where you can find that reepitheliation, cell migration, as well as this collagen deposition in the day 20 from Mashon's trichondrome staining. And finally, we wanted to also understand, like, what is the happening in case of the inflammatory cytokines. So here also you could find that in the day 10 particularly, because we have found that day 10 is actually has a critical timeline to regulate this wound healing. So day 10 actually we could find out that inflammatory IL-6 is actually going down, and IL-10 is actually going up. So pro-inflammatory cytokines are again getting regulated with a dynamic regulation of the anti-inflammatory cytokines. And this is actually regulated by this NRF2 pathway, regulating NRF2 pathway, which is actually found out. NRF2 is a pathway responsible for oxidative stress and counteracting the inflammation. This is also as a published data. So next, we wanted to understand whether combining these two have any role or any additive effect into the wound bed. So the same condition, when we have the burn condition, we have actually used the insulin, eticopride, which is a dopamine D2 antagonist, and together of these two, the topical insulin as well as the a systemic intraperitoneal administration of the etiqulopride. That we have seen that this is the combined treatment, that we have seen that the combination has additive or synergistic effect, which actually exceeds the insulin as well as the etiqulopride if given separately. In the same scenario, we have done the same experiments like with the 10-day and 20-day hematoxylin staining. And so we can find out that this initiation and migration of the cells are effectively going there. And at the day 10 also, we have found that IL-6 and IL-10 that are dynamically regulated, pro-inflammatory cytokines going down, anti-inflammatory these are actually going up. So this is the condition where insulin and etiqulopride has been. When they are together, it can find that it's a very good scenario, good condition, and the synergistic efficacy can be proved from there. Next is like what we wanted to understand is if we take it as a treatment, so what is happening in case of a burn wound patients? So we taken the microarray data from the burn patients, 2 different data sets, which are like and the different burn patients, which has the normal patient and the burn patients together. And we wanted to understand that dysregulated genes here. So the genes going up and genes going down, and this is the common gene for the 2 different type of the data set, 17 and 8. We have put them together and wanted to analyze like what are the role in their in the wound healing scenario. And also we have done protein-protein interaction and followed by hub gene interaction network, which shows the important genes which has some role, and whether these are the hub, like these are the main genes which are regulating this condition. And we have taken there are conditions like the day 10, because we have understood the importance of day 10 particularly. So we have taken the day 10 of these DEGs. And through the pathway enrichment analysis, through KEGG, we could also understand that TGF-beta is one of the most important pathway that is already known, but these genes are actually regulated into the TGF-beta pathway. So among them, this LCN2 was our important, like we wanted to understand the role of LCN2 because it has not much reported anywhere in the wound healing, and particularly burn wound healing. So we could find out that LCN, there is only one report that we could actually come across, that LCN2 is actually a regulator with the MMP9. So LCN2 and MMP9 are complex. They are making a complex, and their MMP9's enzymatic activity also increases. And LCN2 and MMP9 are regulated by STAT3, a major downstream effector of IL-6. So the inflammatory modulation is actually done by the LCN2, which we can actually hypothesize. And this was our hypothesis, like with the wound, there is if the TGF-beta level is increased due to this different condition like a burn, then there is a decrease of the LCN2, which is actually also, accordingly, that decreases the MMP9 activity, which hinders the keratinocyte migration and inflammatory modulation, as well as the remodeling stages. So we wanted to understand, this is a very dirty Western blot. So this is a very last result, we couldn't do much into it. So we, in the same scenario, which in the 10-day burn wound healing, so we took the control insulin treatment, etiqulopride treatment, and the combined treatment. So we could find out that with the treatment of this, when the LCN2 is downregulated, which is our hypothesis, and with the treatment of insulin and etiqulopride both, and the combination treatment that has been restored, that has been upregulated, followed by the MMP9 activity, and which we can actually extrapolate with the faster wounding healing capability of this treatment modality. So this is like here we come to the summary. Like this is an indirect understanding of if in the burn patient condition, what is happening. Like if LCN2 is an effector molecule, and this treatment can, what we are finding in the mice model, so if we can actually translate that into the human scenario, so this can actually LCN2, which is the human counterpart we can found from the database, that can be counteracted, and this can be modulation. It can modulate the activity. So this is like the main summary of my talk. that both the insulin and dopamine has the role in the wound healing. And LCN2 has been a key regulator in the TGF-beta pathway, which can be modulates the inflammation and the migration of the cells, and can be modulated by these two treatment of this dopamine or insulin. And that might actually can be translated into a clinical scenario for further investigation and development of a modality. So with this, I like to thank my students who have done the work actually. My collaborator, Dr. Deeptiman Chaudhuri, who is from Thapar Institute, who done this nano formulation and a few of these formulation works. And I like to thank my director CSI, as well as the funding agencies, which helped me to doing the work. And I'd like to thank you all for being with me and patiently looking.
Okay, thank you, Dr. Basu. Yes, we have some time for some questions. Yes, at the back, I see some hands raising, and then you first, that lady. Yeah. Can you, somebody pass
take
on
the
the
mic,
mic, please? Is it on? Is it on?
The wound healing is different in the diabetic patient. So diabetic patients, like there is a, like I have described there's a continuous inflammatory condition going on. So there is the wound is not healing. So there is a pro-inflammatory cytokines which are actually surging, and that is a chronic wound condition. Diabetic wound is actually that do not heal. In general, wound healing is an orchestrated balance between different phases. When one phase starts, it is actually continued with there is a end of the phase, then continuation of the another phase, and ultimately it is work. So there is a start and stop signal is there. But in case of diabetic wound, if I'm understanding your question right, so there is the inflammatory phase is actually prolonged. So there is a pro-inflammatory cytokines which is surging again and again, and that is actually delaying, and the anti-inflammatory cytokines which is need to come up at the time point, that is not coming. So that is a very hypermetabolic condition in the this diabetic condition.
Thank you so much.
Hello, sir. Good morning.
Good morning.
Firstly, thank you for a great lecture. And my question is similar with the diabetes. So now we are seeing the treatment for type 2 diabetes mellitus patients with the long-lasting insulins, like 24-hour insulins. So these insulins are getting some success in regulating the blood glucose levels, but still the wound healing process is the major concern for these patients. So why do you think these insulins can regulate the glucose levels, but not the wound healing process?
Yeah, it's a good condition because I have mentioned there the insulin we are administering in a systematic, like you're in the bloodstream, and then it's actually acting on the when there's a non-metabolism, you're actually helping in the metabolism. So wound like the one I have mentioned is a topical insulin. So insulin which is localized effect, and that is actually helping in the anti-inflammatory role. So that is a topical insulin, and difference between the systemic insulin that has been working.
Okay, there were two
Thank you.
hands already there, one by one. Yeah, those two people before we close this. Yeah.
Hello, boss, and very informative talk. So my question is that when we are talking here about the diabetic condition type of thing and your normal nanoparticle, which is going to affect the wound healing process, right? What about the people where or the patients where there is a compromised patient, where there is a they're denerved and there's no nerve supply to the wound. So where there this particular process is compromised, or it will be delayed, or whether you have used such groups where the nerve supply is already manifested
In the patients.
or you have used the model, no?
Yeah, we have used the mice model of like there is selective depletion of these nerves that has you can find out there is like the dopamine condition is but we have not checked into this. There are reports I have mentioned. There is reports on that, like if the nerve ablation is done, that will hamper the wound healing process. Even a normal wound healing process, that will be fine.
So it will be different from the neuropathy which is associated with the diabetes?
No, diabetes neuropathy is actually something which is a kind of a pain, is a pain. The TRPV1 receptors are involved, and that is like different thing. But it is like the denervation we didn't check actually.
Yeah.
Diabetes with denervation, that is something different condition.
Yeah.
So there is impaired nerve. Nerves are also impaired.
Right.
So not denervation, there's an impaired nerve is there. The functionality is actually nerve degeneration, axonal degeneration is there.
Yeah.
That is another critical situation. That is obviously part of the diabetic physiology.
Okay.
So we looked into the wound healing part also. But also I know that I work on diabetic neuropathy also. So that is a critical mode situation also. That is different.
Okay, so
Thank
last
you.
question from this section. Yes.
Hello, sir. Thank you for your nice deliberation. So my question is related to biocompatibility of the nano formulation that you have used. So although this was a topical application, so as far as human is concerned, so whenever you are using this, what is the biocompatibility issue? And second thing is that there are the different survival rate for the different types of burning. So people, they are not going to survive if percentage of burning is more there. So
Yes.
in case of your whatever nano formulation is there, if you are going to apply that, so in that case, what type of cure will be there? So this is my question.
So what you have mentioned is actually one thing, like when the percentage of burn, it is very high. So then there is it can be an additive or a kind of a supportive treatment. But if there is so much of burn, so there is, the patient has no chance that way. Like there are few chances if it's a total burn condition. So it's like accelerating the wound healing, which the normal, the burn is there, that can accelerate through this process that we have found. And there are many silverex also, and there are many different types of already medications are there, which can, the patient at the brink of maybe at the very worst condition, that is something different condition.
And biocompatibility.
And biocompatibility, I have mentioned here actually. We have looked into the cell lines in the HECA cell, which is a skin cell, and they are with different time and different concentration. So I have mentioned 60 micromolar of the insulin. That is the total content in this there. The slides also had. I was very fast, so I didn't concentrate much there. So biocompatibility we have checked, of course, the empty this nanoformulation and the insulin nanoformulation together. Both are biocompatible, both in vitro as well as in vivo. We got found.
Thank you, sir.
Okay, thank you very much, Dr. Basu, as well as the audience. And I guess there may be more questions, but I think we can move on. And he will be available during tea time. So
Sure.
I request Dr. Guni to go ahead for the next speaker.
Thank
who
you.
Will
Thank you
be
so much.
online.
So our next speaker for today's session is Dr. Moumita Datta. She's a research scientist at the University of Washington, Seattle, US. So she's actively working on analytical methods to quantify and characterize metabolic pathways and associated metabolites in diseases like colon cancer and liver disease. Her research interests focus on enhancing the processes of drug development. She uses a combination of in vivo and in vitro models along with molecular biology techniques to determine the interaction between environmental factors and pathology of host disease development. Today, she will be talking about microbiome, microbial metabolites, and nuclear receptors, unseen bidirectional crosstalk modulate health outcome. So welcome, Dr. Datta. Also, I would like to mention you have 20 minutes, and after 20 minutes, there will be a buzzer or an alarm. After 17 minutes, there will be a buzzer. So you will get... Okay. And then after 17 minutes, there will be a buzzer indicating that you have 3 minutes to conclude your talk. And also, we cannot see your presentation in the full screen. If you want to change it to the slideshow in the full screen, we are seeing an outline. So I think you're using multiple screens. You need to duplicate. Okay, yeah. Now we cannot see anything. You need to reshare. And also, we cannot hear you. You need to unmute yourself.
It's unmuted, but volume. It will come.
Okay.
Can you hear me now?
Yes.
Yeah, we can hear you.
Okay.
No slide.
Yeah, only background.
Yes.
Okay, yeah, we can see.
You can see now?
Yes, great. Thank you.
Okay. So, hi everyone, good morning, and I am Momita. So currently I am working as a senior staff scientist at University of Washington, Seattle. And I was supposed to be there in person, but due to some complication, I could not manage to be there in person. And so now I'm giving this lecture online. And thanks to Amity University for inviting me to share my research updates to the bigger audience. So today I'm going to share some updates of my current research topics on gut microbiome, microbial metabolites, and how nuclear receptors such as PXR interact with each other and modulate host health and disease development. So before going in-depth discussion, I would like to share some updates on human fecal microbiome. So human gut microbes at this current time is one of the most important topics, and human harbors like more than trillions of microbiome, which play an important role in their health and disease development. And nuclear receptor, there are different nuclear receptors present in human, in us. Among them, like PXR is one of the major nuclear receptor, or also considered as a orphan receptor, which is highly expressed in liver and intestine, and that upregulate the process of drug metabolizing enzyme cytochrome P450 to modify host xenobiotic biotransformation and intermediary metabolism. This gene, nuclear receptor, also play an important role in different cancer, like liver cancer and other metabolic disease development, such as diabetes. So in our previous research, we found that germ-free mice liver represent 90% decrease in mRNA and protein expression for PXR. So that was like really interesting finding to us. So that brings me to my current venture. So before going, as you all know, nuclear receptor is a receptor that stays in the nucleus and directly bind to the DNA after ligand binding and modulate gene expression. So nuclear receptors are two types, generally type 1 and type 2. And today I'm going to mostly talk about the type 2 nuclear receptor, such as liver nuclear receptors. Nuclear receptors such as PXR upregulate the expression of CYP3A, and CYP3A is considered is one of the most important gene that is responsible for the metabolism of 50% of the human drugs from our body. So how PXR works? So PXR binds, it's a heterodimeric nuclear receptor. So when ligand, and there are like different types of ligand today is known, can activate PXR or inhibit PXR activation. Among them, like when the new ligand binds with the PXR, it's it binds with RXR, its activation factor, and work in the genomic DNA and activate expression of CYP3A, which is the basic function of the PXR. So okay. So next, another topic of my discussion is microbial metabolites. And when I am talking about microbial metabolites, I am basically mentioning bile acids or focusing bile acids molecule. So microbial metabolites are different types of compounds that are mainly produced by microbiome. These microbial metabolites are most important. One of the most important microbial metabolites are short chain fatty acids, and literature has shown that short chain fatty acids has a very effective role in different disease. Their upregulation and downregulation can modulate disease progression, and some neurodegenerative disease also impacted by short chain fatty acid. In our case, we consider, as my research interested mainly focus in nuclear receptor and liver mostly. So I focus on bile acid. So bile acid is one of the interesting molecule which is produced in liver as a byproduct of cholesterol metabolism, and then this primary bile acid enters into the gut, and where gut microbiome work on the primary bile acid and produced more hydrophobic secondary bile acid. So this secondary bile acid is basically eliminated, like mostly eliminated from our body through fecal fecal excretion, but some of the bile acids again enters into our liver. So there is a continuous crosstalk going on between human liver, like human or mouse liver and gut, where bile acids and microbiome crosstalk each other with their metabolite production. So, and in different studies, some bile acid are considered as neuroprotective bile acids, and some are considered as neurotoxic bile acid. For example, lithocolic acid or LCA, which is a gut-produced secondary bile acid, is considered as a early biomarker for Alzheimer's disease detection. And DCA or deoxycholic acid is another microbiome-produced bile acid, is one of the marker for colorectal cancer. So these bile acids plays an important role in different host disease production. So this is the pathway of bile acid metabolism in human liver and gut, and the other panel is showing how mouse or murine, gut and liver bile acid production pathway goes on. So my research was specifically interested to understand how this liver gut crosstalk going on, and is there any or more specifically PXR and microbiome crosstalk going on, if there is any crosstalk presence, or through which this crosstalk going on. So in my previous publications and from our lab, we have already shown that different PXR dependent outcomes in host disease development, such as NASH or NAFLD progression increased through PXR dependent way. Some gut microbiome significantly increase or decrease, inflammation producing bacteria increase. So there are several health outcomes we have seen in our previous publications, and now I was interested to understand how this crosstalk going on. Is there any crosstalk or not? So I did some experimental design, and my experiment was divided in two parts. In first experiment, I used mice model, and second experiment, I used clinical samples from healthy human kids donor. So in what's my first experiment was? So my first experiment was using of conventional mice and germ-free mice. And we collected feces, fecal samples, which are the great source of microbiome detection. So we collected feces from different ages and until they are very old, at least 120 days old, and did use multiomics approach like 16S rDNA analysis, metabolite, microbial metabolite bile acid quantification, and we studied hepatic gene expression. And on our second experiment, it was a collaboration with Dr. Sheila Satyanarayan from Seattle Children's Hospital, where we collected human feces samples from this different age group healthy kids. And we again use multiomics approach to analyze the gut microbiome in the developing human population. So what we find from the experiment one, and I would like to remind you, this was our experiment one, where we just compare the gut microbiome difference among our conventional and germ-free mice. So you can see like the alpha diversity was significantly increased when the mice were like old, because their food habit was like increased. They have more food, so there it was very obvious. And beta diversity also showing there are different clustering at different age group. But interestingly, the result we saw when we did some gene expression assay, and we measured CYP3A11 gene. So CYP3A11 is a PXR regulated mouse gene. In case of human, it's CYP3A4. So in case of mouse, it's CYP3A11. And we saw in case of old mice, old germ-free mice, there was significant decrease in CYP3A11 expression. And similarly, secondary bile acid, which I mentioned before, which is produced by the gut microbiome, was significantly decreased at older age germ-free mice. But as those mice were germ-free, it was expected their secondary bile acid production would be none to zero, like none to very less. So that this result was not that much interesting, but we were surprised to see the expression of CYP3A11 in their liver. So we also did experiment 2 and quantify the gut microbiome following 16S rDNA and analytical assay using LCMS. And we saw that there was a, like as expected, as the kids grow older, their food habits change and their gut microbiome also enriched. And interestingly, we saw these three specific microbiome significantly increase in a 3 to 5 years age group kids compared to other. And these three, like Akromansia, Parabacteroides, and Faecalibacterium, these three are very important gut microbiome, human gut microbiome. They plays very important role in certain disease progression, such as IBD and other metabolic disorder. So this finding was really interesting. And we did in our experiment 2, which was from our human dataset, we measured bile acid using LCMS. And interestingly, we saw like as expected, with the increasing age, there was an increased secondary bile acid in feces, and it was interestingly high in case of 3 to 5 years age group kids. Then what was our combined finding from these two experiments? it was that like germ-free mice liver represents 90% decrease CYP311 expression, both in mRNA state and protein expression. And secondary bile acid production significantly decreased in old germ-free mice. Alpha diversity indicates increased microbial richness with age and decreased primary bile acid concentration and increased secondary bile acid concentration through the age. So, and few interesting taxa such as Akkermansia, Parabacteroides, and Faecalibacterium, they increase significantly at 3 to 5 years age group kids. So now it's give me another idea to design another experiment, which we called experiment 3, where we use those 4 age group fecal samples and use fecal microbiome transfer into our germ-free mice and produce them as a eggs germ-free mice. So we used germ-free mice as a control group and use same-aged black germ-free mice and orally gavaged 3 weeks old germ-free mice was orally gavaged with different age group fecal samples. So that, and after 1 month of incubation, we collected tissue from these 5 groups and again analyzed those we collected all sort of tissues and do gut microbiome analysis using 16As. We did metabolite quantification using LCMS and it was targeted LCMS approach and hepatic gene expression study. So again, now I want to remind you that how is the bile acid metabolism takes place in both liver and gut. And here in case of mouse, when after cholesterol metabolism inside the liver, there are two, instead of 2 primary bile acid, cholic acid and skinodeoxycholic acid, there are another type of primary bile acid produced in case of mouse liver. It's called UDCA. And it's considered, like in the literature, it has been shown that UDCA is a very neuroprotective bile acid, which improves some neurodegenerative disorder like Alzheimer's disease. So UDCA is one of the major primary bile acid in case produced in mouse liver, which is not present in human liver. So from UDCA, there are 2 another bile acid produced, alpha MCA and beta MCA. When all these primary bile acid enters in the gut microbiome works on them and produce some secondary bile acid. And besides deoxycholic acid, lithocholic acid, there are some other secondary bile acid produced in mouse model, mouse gut, which are called omega murocholic acid and HTCA. So these are one of the major secondary bile acid produced in murine gut, rat and mice, not in case of human gut. So these are the reminder. So from our interesting from our experiment 3, we interestingly find that similar increase of few taxa such as Akkermansia municiphala, Parabacteroides and Faecalibacterium in case of 3 to 5 group eggs germ-free mice. And there was a significant increase as expected when those mice grows older, like eggs germ-free mice when they have old group human feces. And we measured bile acid in both in different compartments like liver, serum, feces. And among other bile acids, we saw some interesting finding that omega MCA was significantly high in case of eggs germ-free condition. And it's T omega MCA, which is another secondary bile acid, also increased in case of liver. And similar increase was seen in serum as well as feces. So it was very interesting to see like why this 3 to 5 years age group gavaged fecal group is showing that much production of secondary bile acid. Although all the mice was like shared same was incubated in same facility. They had same air, same water and food, autoclaved food. So there was no chance like this group was having some another factor which is impacting this omega MCA production. And we did some Pearson correlation, and it was interesting to see that, as I mentioned, like this LCA, DCA, these all are the like secondary bile acid, and we saw there was a significant correlation with Akkermansia muciniphila over 90% with omega murocholic acid, and similarly other two microbiome like taxa, Parabacteroides and Faecalibacteria, both like all three, they are very strongly positively correlated with omega murocholic acid. And that was a very interesting finding because we saw like when this as in case of our human data, we saw the significant increase in 3 to 5 years age group, and similar increase we saw in our like ex-Jumfrey mice experiment, and we saw similar increase in the like correlation, like strong positive correlation in omega MCA and like this bacterial taxa. Now,
Dr. Datta, sorry for interruption. Could you please summarize?
Yes, I will. So, so we find that in case of germ-free mice, there was a significant decrease in at old age in CYP311 expression. But interestingly, in our ex-germ-free condition, in both liver and ileum, CYP3 expression, CYP311 was significantly high, and that connects the dot. So, and we did the Pearson correlation, and we saw there was a significant strongly correlation between omega MCA in both ileum and liver CYP311 gene expression. We also used in vitro assay, and we used like a HEB liver cell line, HEBG2, and incubated those cell lines with omega MCI and strong PXR activator. Mouse PXR activator is PCN, and human PXR activator with rifamycin. And we saw with omega MCA, there was a significant increase of PXR expression. So overall finding from this data, we saw there was a significant interesting bidirectional crosstalk going on between PXR and microbiome, and this crosstalk is going on through a microbial metabolite which is murin produced and called omega murocholic acid, and this is modulating the whole health and disease development in case our mice model. So in our future goal, we would like.
I think we have to wind up now. You might like to acknowledge the last slide, or that is done.
Yeah, this is the last slide.
Okay.
Yeah, so yeah, this is our overall finding, and yeah, thank you for giving me the opportunity. And yeah, this is my lab and my.
Thank you, Dr. Datta. Sorry for interruption. I know it is quite late, even in the evening. Since we are running late, there are two more speakers in this session. I might like to have only one question if there is one. Anybody? Yes, please. One question. Dr. Datta, please. Yes, go ahead.
Hello. My question is related to the microbiome. Actually, you have seen the microbiome from the gut, and this is basically 1% microbes. Those are culturable. So are you looking for the unculturable microbes also through metagenomics in your study? Are you getting my question?
Dr. Datta, are you here?
She's disconnected.
Oh, she's disconnected.
Anyway, thank you.
There is some technical error, it seems. Oh, here it is. Dr. Datta, did you hear the question?
Yes, I did. So can you hear me now?
Yes.
Yes, most of the microbiome are unculturable. So this is our future goal. We are planning to buy some of these specific like Akkermansia and Faecali microbiome are culturable bacteria. At least we have that capacity. So we are planning to using that approach as well, and also the metagenomic approach. We are planning to do.
Thank you. Thank you.
Mhm.
Thank you, Dr. Datta. So next speaker is Dr. Ashish Gupta. He's an additional director and scientist F in the Pharmacology Division, DEPAS DRDO, Ministry of Defense, India. Trained in biochemistry at CSIR CDRI Lucknow. He has international postdoc experience at Harvard Medical School, MGH, US. His research spans photobiomodulation, photodynamic therapy, wound healing, bioeffects of EMF radiation, and environmental stress physiology with ongoing clinical and translational studies. He has more than 85 publications, a patent, and an age index of 35. He's an NAMS member, serves on editorial board of Advances in Wound Care, and has developed herbal translational products. In today's talk, he's going to talk about translational therapies for non-healing wounds, insights into molecular signaling and clinical evidence. Welcome, Dr. Gupta.
So a very good morning to all of you. And at the outset, I am grateful to the organizers of this conference for giving opportunity to present our work, which has been carried out at Defense Institute of Physiology, Allied Sciences, DRDO, Delhi. So topic is translational therapy for non-healing chronic wounds, insights into molecular signalings and clinical evidence. So I will pass quickly these just initial slide because Dr. Basu has covered all that wound healing is a physiological process, is a highly dynamic, well-orchestrated process, which eventually restores the tissue integrity and functions. And it is fine-tuned by numerous cells like keratinocyte, fibroblasts, endothelial cells, immune cells, which has growth factors and cytokine cocktail, which helps in the healing process. And however, the chronic wounds, they impose a significant medical, social, and financial burdens. And there are so many different etiology and pathology which causes impaired wound or delayed wound healing. Few of them I have mentioned. So these impaired wounds have countless impediment which can be dysregulated inflammation, prolong and decrease angiogenesis, reepithelialization. And there is just a presentation of impaired diabetic wounds. There are so many cofactors or various pathological conditions how which it can cause the impaired wound diabetic. However, in case of burn wound, there is a complete different etiology, and these are the most devastating and prevalent form of trauma. And the normal sequential phase of wound healing is become vulnerable by disruption of epidermal barrier and reduced angiogenesis, sustained inflammation, oxidative stress, and a multifaceted environment of burn milieu restrict the number of treatment available. So based on their etiology, and there are two major factors which governs the wound healing, burn wound healing. First is the extent of burn and then depth of the burn. So depth, on the basis of depth of the burn, it has been classified as a superficial, partial thickness and full thickness burn wound. However, extent of burn wound has been defined on rule of 9. So the translational approach in tissue repair, reparative and regenerative medicines have several challenges in clinical settings, and which requires the search for novel, promising, cost-effective therapeutic interventions. Translational approach rely on combinations of advances in both basic research and clinical understanding. So from bench to bedside approach, and various pharmacological agents like topical growth factor, drug interventions, natural or herbal wound healers, bioactive dressings, which should be a biocompatible, biodegradable, and polymeric composite scaffolds, skin substitutes or graft, smart bioengineering scaffolds, bioequivalent substitutes. So another approach is a non-cellular scaffold, because these are non-immunogenic and there is a decellularized extracellular matrix. And stem cell based therapy, 3D printing, stimuli responsive dressings, which have the factors of pH, oxygen, and temperature sensitivity. There is another modality is that extracellular vesicles, which has been used as a cell-free system for the accelerated wound healing. Beside these pharmacological interventions, the non-pharmacological interventions, which are based on the light-based therapy or electromagnetic field or pulse electromagnetic field. So my focus today is on non-pharmacological therapy, phototherapy, which is delivering of the light, which is a curative use of low-power photons. Besides that, we have also worked on a plant-based bioactive extract, which is basically a seabuckthorn lay berry product, from where we have isolated the bioactive extract from the leaf extract, and we have standardized, and it is a product we have developed based on the various that molecular signaling and mode of action has been worked out in this particular extract, herbal extract, that augments healing of both acute and chronic wounds. It has wide application for skin cuts and scrap, abrasions, incision, excision wounds, and burn and diabetic wounds. And it promotes the USP of this product is that it promotes scarless healing, and it acts as a personal and preventative care. It has various polyphenols and rich in vitamins and phytosterols, which have very important role for combating the oxidative stress during burn wound healing. It enhances the cellular proliferation, collagen synthesis, and it has antibacterial and effects better than available antioxidants, antimicrobial products, and it's safe for dermal applications. So the technology of this product has been transferred to the Amil company, and they are making it as a Heal Master. So now I will shift to that dressings and various pharmacological intervention as has been used, FDA approved Regenerex, which is the PDGF growth factor only, but it is highly costly, and in our country like India and other, we cannot afford such a high cost growth factor based pharmacological interventions. So these hydrogels are having very promising wound healing activity because of they have so many properties, physiochemical and biological activities. So we have also synthesized these bioactive hydrogel dressings using various type of scaffolds platforms, where we have synthesized with the chemical cross-linking, and characterization has been done, and we have compared the these interpenetrating network hydrogels with the available hydrogels in the markets. And with our third approach has used cryogel using freeze thaw technique. So in this, we have incorporated our seabuckthorn active extract, and a patent has been granted on this. And we have found that it has very good healing activity when we have encapsulated this seabuckthorn extract in cryogel dressings. So as I have said that currently the scientists are working on the smart dressings which have bioactive components, like it is nanocomposites or bioresponsive, having components like temperature sensitive, pH, redox, self-healing scaffolds, which has the optimum mechanical strength, controlled drug release, offer dynamic facilitation for these tissue repair. These are biocompatible composite dressing, increase healing process and minimizing the risk of infections. So if there is these stimuli has been given and they they helps in a precision type of wound dressings. Like now, bioengineering scaffolds are beautiful concept, and it has a combination of nanotechnology, regenerative medicines, because it has appropriate physical mechanical characteristics and provides suitable surface chemistry, topography, so that it can mimic. It work as a biomimetic scaffolds, and it offers dynamic control of cytokine cell-cell interaction, physicochemical cues for the product, promoting cell adhesions, differentiation, and proliferation. So these scaffolds have mimic the mimetic native extracellular matrix and it is currently gaining profound impetus among the clinicians. That we have covered this work in biotechnology advances, and it has shown that it has very promising future indications in the tissue repair and regeneration approaches. The stem cells, as I have already said, that they have a potential for growing various type of adult cells, and one of these are the adult mesenchymal stem cells, and second is the human adipose derived stem cells, which are more popular in case of stem cell based therapy for tissue engineering and regenerative medicines. These are these adipose stem cells has abundant autologous options. They can be isolated minimally, and significant proliferative capacity they have, and they are beneficial for wound bed neovascularization and accelerated wound healing. Now I'm shifting to the non-pharmacological activity. Besides these electrical stimulation, the phototherapy has very promising effects, and hyperbaric oxygen therapy and negative pressure therapy and the non-contact low-frequency ultrasound main focus of my talk is the photomedicine. So light-based healing intervention, as it is a potential light-based therapeutic intervention treating various type of disorders, including antimicrobial photodynamic therapy and photobiomodulation therapy. So this antimicrobial therapy is based on specific photosensitizer, which we have select with appropriate wavelength of the light, and they generate type 1 and type 2 reactions, and they generate highly reactive oxygen species like ROS and hydrogen peroxides, which kills very specifically to the multi-drug resistant bacteria, or it can kill cancers also. And even for the blood, that is eye disorder, where the plaque is there, then it destroys that very selectively, because nanocapsulated photosensitizer can be used for a targeted therapy. However, the photobiomodulation is totally rely on this non-invasive, non-thermal drug-free treatment modality for broad applications in the tissue repair and anti-inflammatory pain relief and restoration of cellular functions. And it acts on the cytochrome 4, that is electron chain, that is complex 4, that is it absorbs light and it generate ATP and ROS and nitric oxide. Photobiomodulation therapy can be used with the laser or coherent sources, which are various laser types, and then we can use non-coherent sources like LED, which is a cost-effective therapy. These medical lasers are widely used in dermatology to treat various conditions, including skin resurfacing and cosmetic scar treatment, skin rejuvenation, pigmentation, acne, scar, surgical wound. PBMT can be used in a new therapeutic avenue for many complex disorder, even for neurocognitive, traumatic brain injury and spinal cord injury also. These visible near infrared light has ablative lasers where the property of thermal phenomena is used for treating various disorder. However, my focus is on non-ablative laser, which has low power, and these are used for the wound repair, mitigation of inflammation and pain. Here I'm just showing what is the tissue light interaction with for the photosurgery and phototherapy. So surgical laser, and they have approximate temperature range, and if there is a ablation type of thing and we have to surgically remove, then laser can be used for irreversible damage, photothermal effects. However, in our case, it is less than 40 degree. So this cause the photostimulation of cellular environment, and it cause as a athermal, atraumatic approach for the repair. So the optical window is very important because this phototherapy is primarily wavelength dependent. Here is one analogy that if we are using drug, then there is a name of medicine and doses like 500 paracetamol. We are giving 500 mg for different type of indications. So here the indication medicine is like wavelength. Which wavelength you are using? It could be from visible range from UV up to near infrared. Then it is a medicine. And when we are using those, that is power density or energy density. How much density we are illumination of irradiance, it is very important. So optical window is that from red to near infrared has minimal absorption, lower scattering effects, and tissue components were highly activated with this, and there is a deep penetration ability in tissue compared to other wavelengths. So red and near infrared have showing biostimulation effects. They have biphasic dose response, which shows that there is an optimal dose which has positive effects. However, the under dose has little or no effect. On the other hand, the overdosing could be a negative or little effects. So which is very important. Now, the PBMT involves endogenous cellular chromophores that absorb photons, elicit photophysical and photochemical reactions at a biological scale. Since it's a light-driven approach for healing, so there are so many parameters, like as I've already said, wavelength, power density, and energy density, and mode. Mode is very important because all over the world they have used continuous wave of mode laser therapy. However, very less activity or studies has been done on the pulse mode. The pulse mode are more effective, and our focus on the pulse mode, especially with the these pulse and super pulse mode. So here I am showing that how it is pulse continuous mode, and then there is a on-off in pulse wave, and on the other hand there is a super pulse wave, which are extremely helpful. And penetration depth is showing that the near infrared is penetrating up to dermis and hypodermis also. So these are cellular and molecular actions. We have worked out on this molecular action of super pulse and pulse, and it activated by if we shine the light with these burn wounds or diabetic wounds with the red or near infrared light, the cytochrome C oxidase, which is complex 4, it activated, and it causes brief exposure of reactive oxygen species, which helps in the signaling cascade, and then it also increased PKD, and it affects the NF-kappa B. On the other hand, the complex 4 is also increasing ATP concentration. So our studies has shown that this complex 4 has, beside this, there is another photoacceptor, which is TVRPP 3, that is a transient receptor calcium channel, which has light sensitive activity. And now we have focused that super pulse mode is why so much important, because it penetrate more and it has accumulation of more photon energy in the tissue undergoing for repair process. And during on period, super pulse energy enables quick absorption at cellular subcellular level, while on the off period, the pulse further facilitate an optimal microenvironment for the cell communication, leading to augmented wound healing. Here it is I have shown that if it is a continuous mode, then there is a more flux in case of the super pulse mode. So on the basis of that, we have worked out the molecular action of this particular wavelength, that super pulse 904 nanometer photobiomodulation therapy attenuate inflammatory responses, and it enhances burn wound healing. And we have worked out on the, since I'm a biochemist, so I have worked on the bioenergetic pathways. So it has shown that it increases biological activations and wound healing. So the multi-target approach is another approach where we can combine various combination of therapies, because these are focuses effectively and improve the cure rate of the several diseases, including these impaired wounds. And then combination of bioagents like CoQ10 and superpulse sebuckthorn extract, which have find out in our study with the combination of superpulse and dual wavelength. As such, it can be used as a non-invasive combination of these two treatment, and we have found these multimode therapy is very promising for regulating different phases of tissue repair process, and it helps in the accelerated wound healing. So with the combination of sebuckthorn, we have found that it leads to the reparative properties, and it helps in the scarless healing and fast recovery in third-degree burns. We have worked on these super pulse. Besides super pulse, we have also worked on the pulse 810 and combination. Our hypothesis was that using combination of these wavelengths, it absorbed through the different cellular photoacceptor and varying degree of tissue penetration, which can potentially accelerate the repair process. So the combination of this, I have said that when we are using red light, which is up to 810, it increases mitochondrial bioenergetic pathway. However, in 904, it activated through the TRPV channel, which is light active calcium ion channel. So it increases the dual near and near infrared light synergistically promotes the full thickness burn wound there, and it has our this molecular mechanism validate that it can be further translated to the clinical setups. So here is a depiction that how the various pathways of tissue repair process get upregulated or anti-inflammatory effects have been shown. So basis of this, we have conducted a translational clinical benefits of this super pulse laser, and at Safdarjung Hospital we have burn plastic surgery department. We have conducted a clinical trial based on our findings that this efficacy of this non-invasive near infrared as an adjunct therapy along with the standard of care, that is they have used that gauze dressings and silver sulfidizing cream, and we have delivered curative use of low power photons as a non-invasive adjunct therapy to the burn site. And various grouping has been done, randomization of done in case of clinical trial, and we have done find out the we have taken the blood also and procalcitonin and CRP. Beside this, we have done that clinical visual observation time taken for reepithelialization, wound contraction, and pain assessment. So our study has shown that it has promising wound healing effects, and positive healing outcome could be based on that fast wound contraction, about 40% fast increase, even some in cases only one illumination of light with the super pulse helps in a fast recovery, and there is a 40% fast reepithelialization, and there is a reduced inflammation pain compared to the sham operated which has been treated with the SOC, that is the standard care for the burn patient. So we have published that work in Journal of Biophotonics recently, and so this is the molecular mechanism. We are shining light on this as a non-invasively, non-contact, and it activates the mitochondria and regulate various transcription factor and redox state, and it helps in the cleaning of the wound in a very fast manner. So the key takeaways are that in tissue repair, pharmacological and non-pharmacological interventions are used to reverse the healing complexity associated with the various type of chronic wounds, and these strategy regulates the wound microenvironment, modulate inflammation, promote tissue repair with aesthetic scarless healing. The PBMT is a cutting-edge field in medical science and utilize healing properties of light, interdisciplinary field, which blend the concept of biology, physics, and medicine. So it is a non-invasive, convenient, safe, economical, and it has no side effects. And it is beneficial in reducing pain, inflammation, promoting deep tissue healing, and restoring cellular functions. So the future
Dr.
perspective
Gupta,
of this
Dr.
study,
please wind
yeah,
up.
is the last slide, sir.
Yeah.
So prevailing wound care therapies have demonstrated appreciable efficacy. However, their limitation related to their clinical, translational, and safety, reproducibility, regulatory approval remains. So further research is needed in field of biomarker identification, rapid, reliable, cost-effective bioassay, so that we can make a tailored or personalized medicine for wound care. And integration of digital health solution, multi-targeted combination therapy continue to shape the evolving field of chronic wound care and shifting the paradigm towards the individual to the patient-centric wound care. With this, it is a teamwork, and I'm grateful to the DRDO that they have funded this work, and they have let us work on these from bed to bedside. So thank you with this. If there is any questions.
Yeah. Thank you, Dr. Gupta. So as you know, we are running out, so there can be only one question. Yeah. And rest questions can be during the tea time. No, during tea time. One question.
So actually, my question is that, is there anything which can be used at point of care, like immediately after the burn occurs?
Okay, so based on our and others all over the world, they have used, and some of I have not shown the clinical. So the point of care could be a phototherapy, because our study has proven that we have worked on the second degree burn, but our future study is based on the large scale clinical trial. So you can if the wound is like third degree burn or severe burn, so or traumatic or combat injuries, point of care for our defense soldiers also could be a phototherapy, because it is a handheld therapy and they can carry at the operational field side also. So that could be workout has been shown that it is promoting repair process. On the other hand, it is anti-inflammatory and pain relieving also, because in second degree or second degree especially, there is a ruthless pain, and it's tremendous. Our study has shown is profoundly decreasing the pain because of the work we have, that it reduces COX-2 and substance P receptors also, and it has anti-nociceptive effect.
I mean, traditionally.
Okay, I think you can continue later. Yeah, she had raised an issue question. You can, yeah, please. You can continue during tea time, because I was.
Thank you, Dr. Ashish, for elaborative presentation of your work. What I have seen that you have worked on the herbal formulation, you have worked on the hydrogels combinations, and you are also working on the phototherapy. As per you, and the real work lab you have worked, which therapy is better for the wound healing in the chronic condition?
Okay, so my answer would be that combination therapy, because always there are so many phases. It's a complex process, especially in case of chronic wounds like diabetic, totally different etiology, and in burn wounds or immunosuppressed or pressure ulcer. So etiology is totally different in each and different type of injury. So combination therapy would be the best options. Either it could be combination of pharmacological intervention club with the non-pharmacological, because as I have said that there is a definite role of each modality. So the combination would be the answer.
Yes, the combination is the best one.
Yes.
Thank you.
Yes.
Okay, thank you, Dr. Gupta. And sorry if there is no question and we couldn't, we will go to the last speaker. We started about 20 minutes late, so I'm
Thank you.
reasonably on time. Yeah, thanks.
So I would like to welcome the last speaker in today's session, Dr. Mallika Chatterjee. She's an associate professor, program leader, and chief coordinator of the Neurospine cluster at the Amity Institute of Neuropsychology and Neurosciences, Amity University, Noida. A neurodevelopmental biologist by training, she earned her PhD in biomedical sciences from University of Connecticut Health Science Center, US. She was a Wellcome Trust DBT India Alliance Early Career Fellow at TIFR, Mumbai, and a visiting postdoctoral fellow at Kyoto University, Japan. Dr. Chatterjee received the Fulbright Nehru Academic and Professional Excellence Fellowship during 2023 and 24 for research and teaching at Brown University, US. She also secured the CSIR Aspire Extramural Grant in 2024 for advancing neurodevelopmental research. Her lab focuses on translational aspects of autism spectrum disorder using rodents and fish models to advance the understanding of the disease. Today, she is going to talk about understanding neurobehavior in context of sulfation alterations in proteoglycans across evolution. Over to you, Dr. Chatterjee.
Thank you. Thank you for the kind introduction. And thank you so much to ICoMB for inviting me. It's a real pleasure to speak with the stalwarts here. Yeah. Hi, everyone. And just before we break up for tea, some neuroscience. All right. So I'm going to look at neurobehavior and show you some ongoing studies in the lab and with my collaborators. What you see here is a sagittal section of an embryonic mouse brain. And this is a coronal section. It's a wild-type mouse brain, and which shows green cells, which are GFP positive. This GFP is expressed under the promoter of a transcription factor gene called GBX2, where the story began. Okay, so GBX2 transcription factor is very important for the specification of the region of the brain called thalamus. Thalamus is the relay station, acts as a relay station in the CNS, wherein it helps to filter information from the periphery and sort it out before it reaches to the higher centers of the neocortex. So it's certainly not a very much spoken about brain region, but it is very critical for brain functions. And as you see here with our GBX2 GFP line, during my PhD days, we were able to actually trace what are called thalamocortical projections, that is projections which arise from these green thalamic cells and reach the neocortex in particularly layer 4. They synapse with layer 4 in the neocortex of all mammals. What happens when GBX2 is not there? A bunch of thalamocortical defects rise, wherein in the wild type or control, they would have gone to the cortex, but they get stuck at a region called the IC, or the internal capsule, shown in higher magnification here. And they also crisscross randomly in the internal capsule region. The few cortical axons which reach the cortex from the thalamus are stray. That is to say that they reach the cortical plate aberrantly. But the most prominent defect that we observed and reported in 2015 was, as compared to the control, the thalamic axon, that's the green axons, projected ventrally to another very important region of the brain called the hypothalamus. And this is where our study started, because we wanted to find out what are the molecular mechanisms which lead to this straying of the axons ventrally instead of dorsally to the cortex. At the same time, we were looking at similar mutants which had a similar phenotype, like ventral projections. And the group of Lopez Bendito earlier in 2007, by doing a neat dye experiment, that is, place a crystal in the dorsal thalamus and see where the axons project to. And they saw clearly in the wild type axons were projecting to the neocortex. However, in the mutant, what they saw was a stray of axons were projecting ventrally, shown in higher mag again here. And this seemed to be a similar phenotype to the GBX2 mutant. And thus, we came up with a hypothesis does GBX2 as a transcription factor, as it is known, control or regulate the transcription of certain axon guidance factors? And we propose that ROBO1 and ROBO2 might be downstream of GBX2. What is ROBO1 and ROBO2? ROBO1 and ROBO2 are transmembrane receptors which are present on the surface or of axonal membranes, and they bind to ligands called SLIT. And as a result of that, chemorepulsion takes place and the axons move away from the source of extracellular SLIT. So our hypothesis was, in the absence of GBX2, is ROBO1, ROBO2 expression downregulated? And we did RNA in situ hybridization analysis on sections of the thalamus. And what we saw in the developing thalamic domain, ROBO2, as you see, this is the RNA transcript, which is expressed in the control. It is downregulated, almost gone in the mutant, GBX2 mutant. However, in the case of ROBO1, much contrary to what we have proposed, we saw an upregulation of ROBO1 transcripts in the brain. Now, the ongoing data has already shown from other labs that ROBO1 could compensate when ROBO2 was not there. Similarly, ROBO2 could compensate for ROBO1 when ROBO1 was not there and rescue their function. So essentially, a single mutant would not have any phenotype. All right. However, still we see a mutant phenotype in the GBX2, that is, there is a ventral projection of the neurons. So what explains that? How do we explain that? So what we saw was, originally we know that ROBO binds to the SLIT externally. However, there was a new molecule which was being reported at that time, which was called HSPG, or heparin sulfate proteoglycans. And heparin sulfate proteoglycans are known to interact with SLIT and ROBO and in fact potentiate the protein-protein interaction. So there enters the molecule or hero of the day or hero of the talk, that is heparin sulfate proteoglycans. So a third-party role of heparin sulfate proteoglycan was proposed by us. And to briefly explain what are heparin sulfate proteoglycans, here is the membrane, cellular membrane. You see the core protein of the HSPG, and attached to it is a polymer of carbohydrate residues. Initially, they will consist of glucuronic acids, which are then partially epimerized to iduronic acid. So you have glucuronic acid and iduronic acid, and iduronic acid then gets sulfated at different positions by key enzymes called orthosulfotransferases. And that's what I'm going to talk about today. You have orthosulfotransferases operative at different positions of the carbohydrate residues, which include the 2-ortho, 6-ortho, and rarely the 3-ortho positions. This came up, this created the new concept of heparonome since the last decade. So that we know that protein-protein interactions are very important to a certain cell functions, but we also now know that heparin sulfate proteoglycans can help these proteins to even potentiate their action further. And they can do it in various ways by conformational activation of protein, enhancing protein-protein interactions, co-receptor activation by, like, for example, an FGF receptor activation, or even by concentrating a lot of ligands on the surface of or in the extracellular matrix is done with the help of these sulfated domains. So sulfation of HSPGs has been reported by others to play a very important role in all the different functions. So it's just not the presence of HSPGs, but also their sulfation. So let's remember that information. And let's go back to what is happening in the GBX2 mutant. We did a microarray analysis back in like 10 years back. Okay, so that microarray was in vogue. We did the microarray analysis, and the log fold changes that were seen here we show all the enzymes that are involved in the heparin sulfate proteoglycan metabolism or synthesis pathway and also downregulation. And we saw 3 enzymes, HS6ST1, 2, and 3, which are isoforms present in the mice, and they are all involved in 6 orthosulfotransferase activities. So basically, without them, the HSPGs wouldn't have the 6 orthosulfate group. And we did RNA in situ hybridization of the GBX2 control and the mutant thalamus. And what we saw that there was a complete downregulation of HS6ST1, the enzyme, in the domain or in the mantle domain. That is where the thalamocortical axons first emerge from, and also a complete downregulation of HA6-ST2 in the mantle domain. HA6-ST3 is very lowly expressed, and we've done qPCR to show that it is also downregulated in the GBX2 mutant. However, given the resolution of the in situ hybridization technique, we were unable to detect it in the GBX2 control itself. Going back, okay. So we looked at the double mutant. So we took a ST1, ST2 double knockout enzyme mutant, and what we saw, they partially phenocopied the GBX2 mutant in the case that you see a couple of green axons going to the cortex in the control. However, in the mutant, they are stuck here. We see no axons in the mutant cortex. However, we didn't see a robust phenotype as robust as the GBX2 mutant in terms of the ventral projections. So we can say or conclude that the ST1, ST2 double knockouts were somewhat similar to GBX2 mutant thalamic defects, but not entirely the same. And remember, there's a certain pitfall of looking at multiple mutants, because here in this case, for example, we have 3 isoforms, 1, 2, and 3. We did a thalamic explant culture also, where we showed that the wild-type thalamus, when exposed to, say, a COS source of COS, COS cells, source of GFP, they were not repelling from that source, and they were the neurites from these thalamic explants were everywhere. However, when the COS cells expressed the chemorepellent SLIT1, the neurites are completely repelled to the other direction. However, if you compare this to the double knockout condition thalamic explant, and when exposed to SLIT, there was practically no proximal neurites extended. And the HA6-ST1 and ST2 mutants basically failed to show the chemorepellent effect. That is what we showed in the data here, that the wild type showed chemorepellent effect, while in the knockout, there was no chemorepellent effect. And this is something similar to the GBX2 mutant thalamic axons, that they failed to respond to SLIT2-mediated chemorepulsion. And that brings me to the next part of the topic, or the talk, where we started using a small molecule, which is called sodium chlorate, commonly available cheap molecule. And it has been well established in literature that sodium chlorate at 50 millimolar dosage desulfates at 6 ortho and 2 ortho position. 10 millimolar sodium chlorate desulfates at the 6 ortho position. How? By overall blocking, there is a sulfate donor called PAP, so it overall blocks the sulfate donor. Now, why the interest in HSPG? Because it has multiple implications in disorders, including autism, like social communicative defects has been observed in mice which lack heparin sulfate, and also the upstream enzymes like NDST3 in the HSPG pathway, when they are mutated, the patients show symptoms of schizophrenia and bipolar disorder. Relevance outside neuroscience, well, HSPGs have been shown to have a key important role in cancer metastasis, and many groups are looking for targeting heparin sulfate proteoglycans as therapy. And also in mucopolysaccharidosis, people are targeting heparin sulfate proteoglycans and their sulfations. So that brings me to the zebrafish part. We took 3 chemicals, 3 small molecules, of which I've already spoken about sodium chlorate at 2 different concentrations. Another molecule blocks HS2-ST enzymes, which is basically blocking the 2-orthosulfation, which is called rotlerin, and has not been tested in vivo so far. Trichloroethyl sulfamate blocks the sulfatase enzyme. So it is like a making and breaking. Here you're making the sulfation properties, like adding sulfate is making, and here you're breaking by using the sulfatase enzyme. So basically by blocking sulfatase enzyme, you mean to increase the amount of sulfate groups or residues in the HSPG molecules. However, these 2 are kind of loss of function, because with using sodium chlorate or rotlerin, you're going to block the sulfate groups from being added on to the HSPGs. At Brown, during the tenure of my Fulbright, actually, what I learned, and we set it up in Amity here, we set up a high throughput system by which we can do active behavioral imaging of zebrafish larvae with a very cost-effective approach. So this contains cabinet, which has a DSLR camera, which has 4 proxy plates, as you see here. They are opaque plates. And you can anytime image embryos starting from 5-day-old to 13-day-old zebrafish embryos when they are treated with various solutions, whether acutely or in a chronic manner. And you also have a projector which projects a PowerPoint presentation so that the larvae can basically see at the bottom of the well various presentations, like moving lights, or you can even have a speaker here which emits sounds of certain frequency to give it acoustic stimuli. And how is the data processed? Picture acquired every 6. So the DSLR camera is set to acquire pictures for every 6 seconds. And in total, we have a 3-hour paradigm, and you can have a 2-hour, 1-hour paradigm. You can change it according to your interest. For us, it was a 3-hour paradigm, so it was generating 1,800 images. This is the type of image the camera was taking. And then we used a markerless pose estimation method called Deep Lab Cut, which trained the computer to recognize certain standard larval features like the 2 eyes and the yolk sac. So it was basically like a triangle. And over time T0 till time T1800, it is going to generate and look at the XY, generate the XY coordinates based on your larval features. And this is a machine learning, which we have a code and we have trained the computer. Larval coordinates are detected and a result like files is generated for us. So at the end of the day, we take the results file and we have subtractions and additions by which we are able to generate 25 behavioral parameters in the fish, which includes like 1-hour activity, P15 activity, burst, scoot, which are different types of activity. How does it startle, in response to the acoustic stimuli, etc. And at the end, I'll give you just one example. Here we have used sodium chlorate. Again, as I spoke about sodium chlorate, it desulfates at the 6 and the 2 ortho position, 10 millimolar at the 6 ortho position. This is our paradigm for 1,800 images for over 3 hours. And each has like 10 minutes of binning so that you can average the movement over 10 minutes. And what we do is first 60 minutes, we just don't give any stimulus to the zebrafish. Later, we give a moving red light, upward, downward, and green and blue, and so on and so forth. And then is a pause, and then we 20-second stimuli, interval stimuli, 1 second, and again 20-second interval stimuli. What we saw was that in the 1-hour activity and the P15 activity, 50 millimolar sodium chlorate had a much larger activity as compared to 10 millimolar and 250 millimolar activity. Similarly, for burst and scoot responses, which are greater than, bursts are fast swim patterns, scoots are less slow swim patterns. We saw a similar dose-dependent response of that. We have done that for other molecules also, which include, I will just skip these slides. We have done that other molecules like rotlerin and TCS also. And finally, we created a heat map based on the clustering of similar behavior activity, which showed that molecules at various concentrations of rotlerin and TCS and sodium chlorate clubbed together under different groups, showing that, for example, 50 millimolar sodium chlorate is similar to 10 millimolar sodium chlorate in their activity, but TCS at different concentrations is having a different activity profile on its own. Now, what is the clinical significance of that? We have generated in a similar behavior pattern experiment. We have actually generated, the lab has actually generated in 2024, a drug library they have screened for behavior. It's called a Keyman drug library of 880 drugs. And then with similar behavioral responses, we have mapped our molecules that we tested to these. And what we saw was with a correlation of greater than 0.98. We did a correlation coefficient with a greater than 0.98. Few of them, we saw that they actively map to molecules of D2 and D1 metabolic pathways. Like there were a couple of antagonists, lurasidone, and then you have here aripiprazole, etc., indicating that these molecules that we have, new molecules that we have tested, likely act along the similar pathway of the dopaminergic signal pathway in the CNS, which is not surprising because we have seen a lot of movement changes and differences in startle habituation, etc. And that shows, and there has been literature which shows that there's expression of heparin sulfate endosulfatase in the adult mouse brain, where the enzymes of the heparin sulfate pathways co-express with dopamine receptor. So this is an ongoing study which we want to elaborate further. We have done chronic studies also and compared, they show a different heat map pattern when you compare it with the acute treatment that I just mentioned. And we have set the system up in Amity, and we are in the process of using Fiji already, but we obviously are working on some machine learning with hopefully some of our collaborators to further fine-tune the system. At the same time, we are also looking at reflexes in the rat model. And remember, we are going back to autism. So we are looking at neonatal and postnatal reflexes in various rat models. And our PhD student is currently doing that. So these are the various reflexes that we are checking. And we have a VPA-induced model, which shows delayed emergence of reflexes. And we are going to go back to the fish and correlate with the reflexes that we studied there. Similarly, we observe ADHD-like behavior in the adult progeny of VPA-induced dams. Finally, I'll come here. This is the how are we going to initiate or continue this study is that sugars or heparin sulfate proteoglycans are actually acting as a critical guidance signal, and they help steer the guiding axons. And if you do not have critical genes like GBX2, that disrupts the sulfation, that causing axons to misroute. Based on this finding, we identified some key molecules and small molecules which are potentially disrupting the HSPG pathway. And we did some behavior findings in a high throughput manner on zebrafish larvae, where we could see treated larvae showing significant hyperactivity and altered movement. And we also saw reduced visual response and increased excitability in these larvae. Okay. And finally, as per the existing data around us, we see that autism-like behaviors can emerge from HSPG modulation. And that is a hypothesis which we are going to pursue further in our lab. And thank you for your attention, and to ICoMB.
Thank you, Dr. Chatterjee. Particularly, you were within the time. So we have some time for any question.
Yeah, good afternoon, Dr. Chatterjee. So I think is that when we are doing this high throughput assay,
yes,
was the GBX2 mutant also included in those?
Interesting point, because I plan to do that.
Okay.
I haven't. I just working on the characterization of the small molecules at this point. Yeah, to compare, it would be interesting. Yeah.
Yeah, Dr. Mukhopadhyay. Yeah.
Yeah, a nice presentation.
Thank
Your zebrafish model is very simple model.
Yes, sir.
So you could figure it out that when you treat with certain molecules,
yes,
you see the behavioral change. And I believe that these behavioral changes will be due to change in the signaling pathway. So have you any plan to do the proteomics, genomics, and the transcriptomics to find out?
Sorry. That's a great idea. Eventually, yes. First, sir, I want to look at a dopaminergic, I want to use an antagonist and agonist as a control and see whether I'm actually finding any similarity in behavior to other molecules. But once we have all this out, then definitely we should go into looking at what changes are happening as a result of HSPG.
Then you will find out that what are the changes in the molecular level.
Absolutely.
And signaling pathway.
Yeah, and that would be very interesting because HSPG changes are then causing some molecular changes. That would be something really novel to look at. Yeah.
Good luck.
Yeah.
Any other question from anybody? Yes, please. Because we had some time now. Yeah. Any other after her?
Hello, very interesting talk, really. And my question is so simple, but I wish to ask that how does these heparin sulfate proteoglycans can move around this sympatogenesis so that the neural signaling go well with other antioxidant as well when the therapy is given to those patients that have this behavioral issue?
You're talking about synaptogenesis? Or I didn't
Yes,
get that word.
Synaptogenesis.
And that's interesting because I don't know how there are two types of HSPGs known.
Yes.
One is within the membrane that I showed, and there are others in the extracellular matrix
Yes.
also. Okay, so extracellular matrix molecules, yes, they move around, but the membrane bound might not be that fluid in the terms of interaction. They only interact with the available concentrate diffused molecules which are near the cell.
Yes.
So in coming synaptogenesis, you're relating to what disease person right now?
That's you said autism.
Oh, okay, okay. Yeah. So it is very well known that some of the HSPGs interact with key active zone protein molecules like CNTN, AP2, NR, Neurexin 1 itself, known molecule for synaptic function, is a HSPG.
HSPG.
Yes.
Yes.
Yes,
Okay.
yeah,
Thank you
yeah,
so much.
yeah.
Okay. So if there is no other question, I think we are coming to the end of this session. My apology to all the speakers. First, to thank them, and apology to the speakers and on the audience, because sometimes I have to restrict the time. And with this, I thank everybody and hand over to my co-chair, Dr. Gundeb. Yes.
I would like to thank all the speakers for their insightful contributions. And I would also like to thank all the audience for engaging. And it was a pleasure to co-chair this session with Dr. Malik. Thank you, everyone.
Thank you, sir. Now, on the behalf of ICoMB 26 Organizing Committee, we would like to express our sincere thanks to all the distinguished speakers for their excellent presentation and valuable scientific insights. We now request our respected chairperson and co-chairperson to kindly present mementos to the distinguished speakers as a token of appreciation for their contributions.
Since our first speaker was online, so I think we'll start with the second offline speaker. Dr. Bishwarup Basu, may I request you to please come on stage? Please give him a huge round of applause. May I request Dr. Ashish Gupta to please come on stage and receive his memento. Last but not the least, Dr. Malika Chatterjee, may I request you to please come on stage.
Thank you.
Here, you have to be here.
We
So
would
let's
also
begin
like
for
to
you
take
also.
this opportunity to honor our esteemed chairperson and co-chairperson for their guidance and support throughout the session. We request Dr. Amitabh Mukhopadhyay and Dr. Vijayan to kindly honor the chairperson and co-chairperson.
So please put your hands together for our chairperson, Dr. D.N. Malik. So once again, we thank everyone for making this session a success. We now proceed to the next segment of the conference, and that is that we are breaking up for the most awaited tea. So there's an announcement. The tea for delegates and registered students is in I2 foyer. For faculty, it is in F1. And for invited speakers, it is in I3 lounge. Right. Yeah. So we request all of you to please come back after high tea to the same venue, as we have a very interesting session lined up. That is industrial approach to medical biotechnology, where we have list of many industrial professionals who will be sharing their valuable insights. So all of you, please join back in another
15 minutes.
15 minutes. Thank you.
Good afternoon, everyone. We are starting with our session 14, which is titled as Industrial Approach to Medical Biotechnology. Basic research finds its true meaning only when it moves beyond discovery to application. No scientific inquiry is truly complete only when it is translated to solutions that benefit society. Industrial engagement plays a crucial role in vital journey from knowledge to impact. So this session is actually meant for that only. For this session, we have a privilege to begin with introducing the chairperson for this session,
Professor Dr. Simran Tandon from Amity University, Uttar Pradesh, Noida. May I cordially invite you on stage, ma'am? So Professor Simran Tandon is currently serving as a Director of Research at Amity Institute of Molecular Medicine and Stem Cell Research. Her research focus lies in the area of disease biology, with a special emphasis on breast cancer and kidney stones. Her lab is dedicated to unraveling the molecular mechanisms underlying the therapeutic potential of alternative systems of medicines, such as homeopathy, in the management of these diseases. Dr. Tandon has secured funding from various government agencies and has published her research findings in leading scientific journals. Her research group aims to contribute to the development of novel therapeutic strategies against breast cancer and nephrolithiasis. Please put your hands together to welcome our chairperson, Dr. Simran Tandon. She is accompanied by co-chairperson, Professor Dr. Tanveer Naved, again from Amity University.
Dr. Tanveer Naved is serving as Deputy Head, Health and Allied Sciences at Amity Institute of Pharmacy. He is an accomplished academician and a researcher with expertise in pharmaceutical sciences and allied health disciplines. Dr. Naved has been actively involved in teaching, research, academic administration, and curriculum development. His scholarly interests span interdisciplinary areas of pharmacy and health sciences. He plays a key role in fostering academic excellence, research culture, and industry-academia collaboration within the institution. Please.
Please give a huge round of applause and welcome him. So we are officially handing over to you now. Thank you.
You, Master of Ceremony, for nice introduction. So this is the 14th session of this conference and the last session, but it focuses on the industrial approaches to the medical biotechnology. I like to invite the first speaker of the session, Ms. Shobha Iyer, the Senior Manager, Quality Assurance from TSSS Takara BioIndia Private Limited, who will speak on the topic of DSS Takara BioIndia Manufacturing Excellence. I request Dr. Ms. Shobha Iyer to please come. Thank you.
Good afternoon, everyone. I am Shobha Iyer, and I'm working as the Senior Manager Quality Assurance with DSS Takara Bio India Private Limited. Thank you very much for providing us the opportunity to present our company in this esteemed conference. The title of the presentation is DSS Takara Bio India Manufacturing Excellence Going Local. So before I start with the presentation, I would like to take a minute to emphasize on the term global, which is the theme of my presentation. So the term global is basically a combination of two terms, global and local. It essentially means establishing production systems of world-class standards, but considering the local customer needs and preferences. So with that, the agenda of the presentation, I will give a brief overview of our company. The manufacturing excellence, a range of products manufactured by DTI, and custom bulk and OEM capabilities of our organization, and finally, a summary slide. So DSS Takara Bio India Private Limited is a joint venture which is established between Takara Bio Japan and DSS ImageTech India Private Limited in the year 2011. Each company individually has its own area of expertise. While Takara Bio has been a global leader in the life sciences field with providing the reagents which are required for research in biotechnology, DSS ImageTech's expertise lies in providing cutting-edge solutions to researchers and clinicians in the various fields of healthcare. So this slide presents the various manufacturing locations of Takara Bio Group. So we are spread across the continents. We have manufacturing facilities in the US, Europe, Asia, and in India as DSS Takara Bio India Private Limited, which we also refer to as DTI. So the objective of setting a manufacturing facility at specific location is also our business strategy to build a local manufacturing system based on a balance ensuring improved efficiency and risk reduction. In India, if you look at DSS Takara Bio, then the primary objective of setting a manufacturing facility here in India is to manufacture and market the Takara Bio research kits and reagents in order to adequately meet the needs of the Indian scientific community. We are probably among the only MNCs to have a manufacturing facility in India, thus contributing to the Make in India mission. The Takara Bio Group brands, if we talk about, we have the Takara brand, CloneTech, and Cellatis. Again, each of these brands have originated from a separate company with their areas of expertise. So if we talk about the Takara brand products, we offer a wide range of biotechnology research kits, molecular biology-based reagents. CloneTech, more of the gene expression related reagents, then Cellatis, products related to stem cells. Apart from this, we are also associated with several international prominent brands like Makri Nagal, which offers a complete range of nucleic acid and protein purification products. Promosul, the products are basically the cell-related media and cell lines, and TriLink, which has products related to a special focus on the messenger RNA-related products. So with this invaluable experience in manufacturing the products and storage, preservation, and also the shipment of products in order to ensure that these products reach in the right quality to the customer, we have also come up with our own indigenous brand, that is DTI, wherein we design, develop, and manufacture products according to specific customer needs. So as you know that India is on the verge of biotech revolution, and several policies are in the area of biotechnology, particularly to kind of strengthen our position as a global bioeconomy leader. So there are several policies in place, including the DBT National Biotechnology Development Strategy. Broadly, if you look at the goals of these policies, it is to build a strong research academic partnership, ensuring quality assurance of Indian products as per the international standards, and creating an environment for the development of biotechnology to a higher level of growth. So indigenous manufacturing of kits and reagents is extremely crucial for the successful implementation or execution of such strategies. The benefits, of course, include strengthening self-reliance, faster localized product customization, cost efficiency, and global competitiveness. And of course, with this, we hope to put India we hope to enhance the image of India in the global scenario. So this is a glimpse of our manufacturing facility. We are certified to meeting the ISO standards of ISO 9001, which is a generic quality management system standard, and ISO 13485 standard, which is a quality management system, but specifically for medical devices. So the features of our manufacturing, of course, we are indigenous manufacturers. So we are in line with the Atmanirbhar and Make in India mission. We are manufacturing under conformance of internationally recognized standards. Our facility is equipped with excellent infrastructure, including a class 100 clean booth, which is where we manufacture all our products. And since most of the products require a -20-degree storage temperature, we have several freezers in our facility, which are connected to automated temperature monitoring systems. And each lot that is manufactured it undergoes a robust quality control process, and each lot is verified before it is actually available for sale. So coming to the range of Takara Bio products, of course, when you talk about Takara Bio Japan or the company, we have a huge portfolio. But in India, we have selected products that are more required in our scenario. So we do have products ranging from nucleic acid extraction kits to PCR enzymes, the RT-PCR enzymes, the ready-to-use premix format of PCR and RT-qPCR. Endpoint-based PCR, real-time PCR, then cloning kits, competent cells, general laboratory reagents like DNA ladder, protein markers, agarose, and so on. We also manufacture glycerol-free enzymes, which are particularly suitable for if your downstream application is going to be the lyophilization of the product. So as of 2025, we have over 120 products which are available in market. We can see that we are covering through the categories of all the categories in both our brands, Takara brand as well as the DTI brand, and we're gradually increasing the number of products to be added. Of course, we are constantly monitoring the customer requirements, and based on the requirements, we plan to design and develop these kits under DTI brand. So I cannot be talking about every single product today, but I would like to just highlight some specific examples. So when we talk about the PCR enzymes, we are manufacturing the routine PCR enzymes, as well as if your need is you need a high speed and a highly accurate enzyme, then we have the Prime Star series. So this is some of the data which I am showing. Again, this is just a representative data of some of the products. You could find the details of every single product in our website. So just to show you that when we're saying PCR enzymes and we have 4-5 different enzymes, what is the difference between the enzyme? So it basically depends on the need of the customer. For example, this image is trying to show the PCR amplification of a target region, difficult target region, a high content of GC. And you can see the 1 and 2 where a normal routine Taq polymerase enzyme and a routine buffer is used, there's no amplification. But you can see in lane numbers 3, 4, 5, 6, where we've used our LA Taq along with a GC specialized buffer, which is also available as a catalog product. We can see good amplification in this product. Similarly, this image is a comparison of our product, Takara EX Premier, with the KOD, which is of Toyobo. So we can see for be it a highly, high GC content or a high AT content, a smaller amplicon, a long amplicon as long as 4 kb, our product is able to amplify this well in comparison to an equivalent kit of the competitor. This is an example of R055A, which is suitable for ultra long sequences. So we can see that it is able to amplify right from 0.5 kb to up to 53 kb. And of course, we do have comparison results of these as well. Again, this we can see that our product Primestar GXL is able to amplify high GC content in comparison to other companies. Then we also have the qPCR range of products. We have the intercalator-based, probe-based, one-step RT-qPCR kit, two-step RT-qPCR kit, and this is just a representative data of our TB Green series intercalator-based product, where we're showing that it's highly specific compared to the competitors, where out of the 32 genes, we are not seeing any kind of non-specific amplification here, but it's detected in the competitor. Similarly, a product which is highly resistant to inhibitors. So in case you're working with direct samples, then this product may be much more suitable. We do have other cloning products, vectors, and chemically competent cells. I'm not going into much detail, but these are the images of our products. When I'm just showing the images of the packaging, it's just to emphasize on the fact that all the packaging, we have performed the necessary validations, be it the primary packaging or the secondary packaging, in order to ensure that the product integrity is maintained up till it reaches the customer. Coming to our custom capabilities, so the 120 products that I mentioned, those are just the off-the-shelf catalog items, but there is always sometimes there is a need that a customer requires just the enzyme, but not the entire catalog product, which may include buffers and other things. So in such cases, we are open to such custom requirements where we can just provide the enzyme as it is. If there is a requirement of higher volume of the enzyme, like standard pack is giving just 250 units, but you need 1,000 units of the enzyme, then we can provide such changes. Other formulation related changes, most of our products are in a 2x concentration for our routine PCR assays. But if there is a requirement of a higher concentration master mix, like a 5x or 10x to perform multiplexing or to accommodate the higher volume of templates, then that can also be accommodated. So the process is very simple. Based on the customer need, they share the detailed requirement. We'll contact with the customer to kind of understand their requirements very clearly before we begin the design of the product. We manufacture the test lot. We also perform our internal analytical evaluation just to confirm that the design is okay. Then we provide the sample to the customer for feedback. Parallelly, we keep the samples for stability studies to kind of understand how long the product is going to be stable. And then if the customer feedback is okay, we'll provide them the required volume. So at all stages, it's very important that we kind of ensure that we get the right feedback from the customer, and we are open to listening to their feedbacks and accordingly improve the product. So just to summarize, at the Delhi facility of DSS Takara Bio India, we are manufacturing products both under the Takara brand as well as the DTI brand. We are manufacturing it under the conformance of QMS of 9001 or 13485. And we have the capabilities of customizing the product as per your specific requirement. So just to end, we would like to contribute to the development of molecular biology in India by driving superior research with our high-performance PCR enzymes, and also by being enablers to IVD kit manufacturers to develop reliable diagnostic assays and consequently to the health of mankind. That's all. Thank you.
Okay. Very nice presentation, Shobha, and it was really nice to know all the initiative by Takara India, especially the new age PCR technology. So I request the audience if any quick query or question relevant to this presentation, please. Yes.
Peptides. We are not into the manufacturing of peptides.
Okay, okay. No worries. Thank you.
So thank you very much, Shobha.
Thank you.
Now I request the next speaker of the session, Mr. Akhil Gupta, Marketing Manager, Life Science Group, Bioride Laboratories, and he will speak on next evolution of droplet digital PCR. Seems a very interesting topic.
Hello, everyone. Good afternoon. So, thank you very much, first of all, to Dr. Banshri for giving me this opportunity to present over here, come and interact with you. My name is Akhil. I'm marketing manager from BioRed Laboratories India Private Limited. Quick introduction about myself. I'm double alumni of Amity University. I have done my integrated MTech from AIB only. And today I'm here to share with you about some new developments in the field of PCR technology. So quickly, I want to know from the students, how many of you know about PCR and qPCR, what they are? Quick. Some hand raise. Only few? Okay. No problem. But again, this is quite an important domain of molecular biology of PCR research, where I'm gonna talk about 3 different era of PCR technology. Okay, so first it came in the form of conventional PCR, okay, where we used to do the DNA amplification and see the results on a gel, right? So that was a qualitative analysis, which used to happen or still being done with a conventional PCR method. Then came real-time PCR or qPCR in 1993. That was the second generation of PCR technology with which we can get relative or indirect quantification. Okay, so where in this particular method or technology, we use standards and references to get quantification of our target. Then in 2011, new revolution came in PCR technology with digital PCR. So in digital PCR, what's the benefit? We are actually removing the need of any standard or reference. We are able to get absolute quantification of our target in a faster way. And how we are attaining it? With the help of partitioning. So what's the reason behind this partitioning? So we have a bulk reaction in PCR or real-time PCR. In case if there is any rare allele of interest, there is a high possibility of missing that. Just like if I ask you to find a needle in this haystack, it would be difficult for you just by looking at it, right? You have to go deeper in the details to find out that needle in this particular haystack. However, if I segregate this particular haystack into smaller parts, it's quite easier to find out my needle, which is hidden in this particular haystack, right? So in the similar way, what we do, we have a bulk reaction, right? And we partition those particular reaction components into 20,000 different partitions, with which we have the capability to detect even a single copy of that rare allele in the PCR reaction. The process of digital PCR is quite simple and similar to that of qPCR. Again, we prepare the reaction where we have our DNA, we have our master mixes, right? So we have a similar reaction setup in digital PCR. After that, what we do the partitioning of that reaction mix, and then we do the PCR amplification, right, with the help of a thermal cycler. And then we read those particular partitions to see the number of partitions having our target of interest, with which we are able to quantify, or I would rather say, absolutely quantify our target of interest. And in today's scenario, we are also able to do multiplexing, which means that you can analyze multiple targets simultaneously in a single well of a PCR plate. With digital PCR, or rather I would say droplet digital PCR, which is a proprietary technology from BioRed, we can perform so many different types of applications on the same platform, whether we are talking about SNPs, indels, fusions, CNVs. So different type of variants can be studied simultaneously on the same technology with high level of sensitivity and precision. Giving a brief background about BioRed, we have an expertise of more than 35 years. In fact, we were the first one who introduced the PCR platform after the introduction of the technology way back in 1980s. And since then, we have been front runners in introducing different types of PCR methods or platform in the market. So when we talk about digital PCR, we were the first one who introduced this technology in the form of droplet digital PCR in 2011. And since then, it's been like 14 years of continuous research and innovation where people have been able to release more than 12,000 different publications with this technology. And on the same platform now, we have more than 5 lakh different assays, which can be used directly by different user groups for their research as well as for clinical purpose. And now we have different type of instruments which can help users based on their research needs, not only applications, but also on the sample throughput, where, as you can see in this particular image, the journey started with 2012, QX100 platform was released, and in 2021, we introduced QX600, which you can multiplex up to 12 different targets in a single well. Now, as I mentioned, there has been continuous development in this field. People have been requesting for more and more markers to be incorporated in their panels, whether they are working on research or diagnostics. So they have continuous need for advanced multiplexing with which they can save on time, they can save consumables cost, and also they can reduce the sample input for their application. Looking into consideration of that particular requirement, we at BioRed have introduced QX700 platform, which has been a revolutionary platform, providing up to 7 different target multiplexing in a single well. And also it's fully automated, where any person who has expertise onto qPCR can simply transition onto this platform of droplet digital PCR, and they can easily perform experiments from 1 to 384 samples in a single run in an automation friendly manner. We have 3 platforms in this range of QX700, where we have the basic platform from 48 sample processing capability, ranging up to QX700 HT, where you can process 384 samples in a single run. And all these platforms allows you to do multiplexing of up to 7 targets in a single well. Another capabilities, as you can see, you can finish the protocol as low as in 2.5 hours, which is even faster than any of the real-time PCR protocol time. And the process is quite simple. If you can see, we just prepare the sample in the form of reaction mix, load it on this particular chip, which is just a process of 15 minutes. Then the protocol is of 2 hours on the instrument, and then we do the analysis in approximately 15 minutes. So the entire workflow from sample prep to data analysis can be finished in as low as 15 minutes, 2.5 hours. Talking about the consumable, we have this particular chip. Again, it's quite easy to work on these, as these are automation and multi-channel pipette compatible. We can simply load anywhere from 1 to 16 samples on a chip. You can run multiple PCR programs simultaneously, And with the higher throughput platforms, you can process as high as 768 samples in a day. So when we talk about applications, because again, when a platform is launched, it is always considered to be a multidimensional utility asset where different departments or different, I would say different multidisciplinary research segments can adopt that particular platform. So whether we are talking about microbiology research, basic biotechnology, biopharma, or applied testing, this instrument has got utility in all these particular segments. So that was a quick introduction on the technology. I know it might be difficult for you all since you are in the basic research right now, but definitely in future, you might be able to utilize or work on this particular technology of digital PCR or droplet digital PCR. I would like to leave you all with this new offer, which we have introduced for all our researchers in this month. This is a special offer on our instruments and consumables where you can take a benefit of up to 50% off on all the consumables from BioRed. When we are talking about amplification reagents, plastics, Western blotting consumables, gel buffer and reagents, and also you can avail up to 65% off on major BioRed instrument and gadget combos. So it's definitely a good perspective when we are talking about basic researchers. They require help in day-to-day research, right? So this particular offer is aimed to provide that particular benefit to all the budding researchers. So I would like to thank you all for giving me this opportunity once again and wish you all the best for a great future. Thank you very much.
Very nice presentation, Akhil, and we always feel proud on a distinguished alumni like you. So I request, yes, one question.
There are a few parts to this question. First, what is the amount of template required?
So again, this particular technology has a capability of detecting even a single copy of target molecule, even a single copy. And if you are talking about the starting sample, right? How much is the DNA input, basically? So some of the assays even can go up to 1 nanogram or 1.5 nanograms. So that's a starting input in terms of that.
So,
So it depends on assay or kit on which we are working.
So you have to use the whole full chip because in qPCR, you can you don't have to use the whole plate.
Yes.
So why should we choose digital instead of Q?
So again, when it comes to sensitivity, as I mentioned, we have the capability of picking up a single copy, which might be difficult in qPCR. So this particular technology, because again, as I mentioned, we partition our reaction mix, right? So these are different micro chambers in which you are doing 20,000 PCRs in parallel, basically in those partitions. So with that, we get a capability of detecting even a single copy of a mutation of a rare or any low express gene. So that's why it is much more sensitive in comparison to real-time PCR. And again, we don't require any standard or reference. So again, if you are using qPCR in any experiment, even if you are running one sample, you have to process your standard and control every time. But in this particular method or technology, you don't require that.
What is the cost of the machine?
The platform costs somewhere around 70 to 80 lakhs. So capital investment is heavy, but the per sample cost, processing cost is as low as 500 rupees.
It is 4-5 times the qPCR.
Yes.
4 times at least.
4 times, yes. Agreed. So capital investment is more in comparison to qPCR. Chip PCR is different. This is more of, there are different methodology of doing PCR. So this is slightly different from that.
Okay. Actually, chip PCR, maybe the solutions and all those things need not to be added.
So again, there are different variants to it. There are different methodologies where the consumables are preloaded on the chip and then PCR is performed There are different point of care testing devices also. So those are more on the chip PCR based methods.
And you told that simultaneously many genes can be.
Yes, with the help of multiplexing.
No chance of false positive.
No, because the primers and probes are specific for that target, right? So you are only getting specific signals where amplification is happening with that specific combination of a probe and primer. So there is no chance of many false reporting over there.
Okay. Thank you.
Thank you.
Yeah,
There
hi,
is a
Akhil.
question from sir. Yeah, sir, please.
Myself is Amitabh Mukhopadhyay. I have a close association with the BioRed.
Right, sir. Thank you.
Years. Hello. Yeah, I have a close association with BioRed for last 25 years. Even when Ronnie was the country
Yes.
manager. And almost all the officials and others knows me very well. I have two things to clarify from you. One is that the offer which you are talking about, that 60% on instrument or 65-50%, that offer is for certain period of time or
Yes.
just open?
This is for specific time from 1st of January to 31st of March. So you can avail.
So basically the financial year closing sales.
Yes. So that's why the offer name is end of year offer.
And the other thing which I have discussed earlier also even with several other officials, you can look into a possibility which is done by several companies across India and even Zambia and other places. So you can think about it. We can give you a place where you can put your high-end instrument, and that facility, our faculty can use it on time basis and divide the time. So we can use your instrument, of course, with our reagent. And the dual benefit for this, that we can use your instrument, of course, we'll buy the reagent for that. And your benefit will be, you can use the same room or facility, which will be probably able to provide or will provide, that can use in a time-bound way, divided time. You can also use it as your training centers, your customers and others. So that will be a dual benefit for you as well as us. So I'll appreciate that if you can think about it and let us know.
Sure, sir. Definitely. Thank you.
Thank you. Thank you so much.
Thank you.
Our next speaker is Dr. Surbhi Gupta. She is the application scientist in the Life Sciences Division of Horiba India Private Limited. And the title of her talk is on ViewSizer 3000, nanoparticle size measurement through visualization. I would please welcome Dr. Surbhi.
Thank you for the introduction. Good afternoon, everyone. I am Surbhi Gupta, application scientist at Horiba India, Bangalore. Horiba India, Bangalore. So today I am going to present ViewSizer 3000 nanoparticle size measurement through visualization. Before starting with the presentation, let me introduce to Horiba. So Horiba is a global leader in scientific instrument with its headquarters in Kyoto, Japan. It was founded in 1945 and incorporated in 1953. Current chairman and group CEO is Mr. Atsushi Horiba. So the Horiba is consists of 47 group companies in 28 countries, showing its strong presence in the globally. So it is divided into 3 segments: energy and environment, bio and healthcare, materials and semiconductor. So Horiba offers diverse range of particle size analyzers based on different techniques. like LA 960 and LA 350, these are based on laser diffraction, whereas SZ100 and View Sizer 3000, they are based on DLS, dynamic light scattering. So as we know that this laser diffraction and dynamic light scattering are the widely used technique for the particle size distribution. However, with these techniques, we won't be able to get a very good resolution, high resolution for the particle size. So, as we know that polydispersivity is a well-known problem, as in a polydispersed sample, large particles scatter a lot more than small particles, making it difficult to detect or track small size particles. And it is discussed in ISO 19430, particle size analysis, particle tracking analysis PTA method, which states that sample polydispersivity affects the ability to track and therefore analyze different size fraction in particle number size distribution. So with the Horiba's View Sizer 3000, we integrate simultaneously three lasers with different powers to accurately measure size distribution in a polydispersed sample. So here you can see that on the y-axis, it's a scattering intensity, and on x-axis is the particle size. So you can see that that the scattering intensity is of order 8 to 10. There's a variation in 8 to 10 order, whereas the size variation is in second order. So if your particle is scattering is large, so scattering intensity is much more, and it will mask the small particle. So in the View Sizer, we have three lasers with different power. So red laser, green laser, and blue laser. So the low power laser is red laser, and then medium power green laser, and high power blue laser. So with this and there is a video camera which record track of every particle, and then software is used to detect the. So here this is the video from the View Sizer. Let me show you this video. I'm not able to play this video. So in this video is from the View Sizer, and you can see that so the three laser works like so if the particle is very large, then this red laser will work, and it will so you can see that there is nice spot in blue image. So in this case, what happens that the high power laser blue is used, and it will detect the scattering from the small particles. And when the particles are very large, so you can see here that the blue image, and there are large blobs over here. And whereas when you tag these particles in the red laser, you can get the very nice spot in the red laser. So then, so this is the video for that. So here you can analyze all the wide range of nanoparticles simultaneously with a very good resolution. Apart from accurate and reproducible measurement of highly polydispersed colloids, the traditional particle size analyzer faces many challenges. They will not estimate particle number concentration, particle kinetic processes, and will not visualize particles. Whereas in View Sizer, you can visualize each particle. So all these challenges can be overcome by View Sizer 3000. So it will cover the particle range from 10 nanometer to 15 micron, and it can measure particle concentration in the range of 10⁵ to 10⁹ particles per ml. And we can also track the movement of the particles, and it allow optimization of kinetic processes. And apart from that, there is another mode in this, is the fluorescence mode, that I will discuss in the later slides. So this is the optical path of the view sizer. Here on the left, you can see there are 3 lasers, and the laser from these laser source, they are focused on the sample in the cuvette, and then the video camera, colored video camera is used to record the movement of each particles, and then the software track the movement of each particle, and then it will choose the best data for each particle. So this system comes with a cuvette-based assembly. The advantage of having this cuvette-based assembly is that it is easy to clean cuvette, which as compared to the flow cell. And this system is also comes with a insert, which is a Horiba patent insert. So the advantage of this insert is that it will only allow the Brownian motion, and it will arrest all other motions. And it will stabilize the sample to avoid drift, and also reduces convection effects. It also comes with a stirrer, so that you can, in between the video, you can make your sample homogeneous, and you can record multiple videos of the same sample, but you are not analyzing the same volume every time. So the the fluorescence, it comes with the fluorescence mode. So in this, what you can do, if you have a mixture of 3 particles, suppose A, B, C, then you tag your one particle B with a fluorophore dye, then you can measure only that particle with this using long pass filters. So in the normal mode, all the 3 lasers are on, and then you can detect all the particles from in that mixture A, B, C. Whereas in fluorescence mode, what will you do? That if your particle B interacts with the blue laser, so in that case, you will turn off other 2 lasers, green laser and red laser, and then the scattering from all the 3 particles is measured, but only B particle will emit the light, which is of longer wavelength. Then this filter is used, which block all the scattered light, but only allowed to pass longer wavelength light, which is from the particle B. So that you can, if you have a mixture of 3 carboxylate fluorescent beads, so without fluorescence mode, you will see this, all the particles will be analyzed, whereas with the fluorescence mode, only red with the red laser, or only with the green laser, or only with the blue laser will be identified. So I will go to the applications of this. So view sizer can be used to measure the aggregates in the protein, to measure size of liposomes, exosome, extracellular vesicle, viruses, and vaccines. So I will directly go to the various applications. So here, this is the case study where you can see that lysozyme is heated in a PVS buffer. It is heated to 60 degree So you can see that even the buffer is heated. You can see there are no particle in the PBS buffer after heating. Whereas in the lysozyme before heating, there was no particle and the screen is black. Whereas after heating, you can see there are the particles aggregate. These are the aggregates up to micron size. It can also be used in the characterization of extracellular vesicles and exosome. So view sizer can be used to count exosomes or extracellular vesicles. As we know that the increase in exosome population may be linked to onset of disease, and this fluorescence mode can be used to measure only the exosomes from the other particles of similar size present in biological fluid. So here is an example. You can see so the exosomes are derived from the human serum. In the normal mode, you can see that the total exosomes can be the concentration of total exosomes can be measured. Whereas if you label one particular exosome in the human serum, then you can quantify the fluorescently labeled exosomes using fluorescence mode. It can be used to study viral vaccine in terms of stability, purity, and total viral concentration. It can also be used to study downstream processing of viruses to optimize virus yield and quality. So here is a case study where view sizer is used an orthogonal tool to plaque assay and qPCR. So in this, we have shown that there is a good correlation from the view sizer, particle count from the view sizer to the plaque assay. So the plaque assay takes days, whereas you can do this with the view sizer in 15 minutes. So from this, we conclude that view sizer is a powerful tool of examining polydispersed sample with high resolution distribution by characterizing individual particle. It enables accurate particle count, concentration measurement without the need of any calibration standards. Apart from the particle size analysis, it can be used to visualize particles and processes to analyze fluorescence particles of the biological samples, and it also allows optimization of kinetic processes. Thank you.
Thank you, Surbhi. Very informative lecture on the applications. We just have time for one quick question. Anyone? Okay. Thank you so much. Thanks a lot. Thank you. Our next speaker is going to be delivering a talk online. He is Mr. Ranjit Chaudhary, Senior Product Applications Specialist at Carl Zeiss Private Limited. And he will be talking on Discover the Dynamics of Life with Optical Sectioning Tools, Volume Imaging.
Online connected.
So, good afternoon. I am Ranjit from Carl Zeiss India. First of all, very thank you to MIT University and ICoMB for giving this platform. So we will deliver a presentation on optical sectioning tool, and majorly it will be volume imaging. I have my colleague joined online, Mr. Suparna Gupta. He is our application specialist. So he will take you through the presentation. Thank you.
Thank you, Ranjit. Am I audible?
Yes. Yeah.
Thank you. Thank you, Ranjit. Thank you, everyone, for giving this opportunity for this particular talk. So, as we discussed so many things about the genomics part, so now slightly towards the, we can say, cell analysis or about the life through images, how we can do the things. And when we talk about the images and all, yes, always microscope is one of the tool that we are using on our day-to-day life. So when we think about any kind of biological application, so yes, the fluorescence imaging is one of the major part that we are using on our day-to-day life, starting from your fixed sample imaging to live sample imaging. And when we talk about the live sample imaging, we are talking about the dynamics of the life. So as the biological samples are mostly 3D in nature, and that's why the optical sectioning tool, different optical sectioning tool plays a vital role there. And the idea here is how we can capture the volume, entire volume in a quickest way. And if we see the entire workflow in terms of different type of sample type, so it starts from cultured cells. And for getting the more close relevance, so yes, different type of models we are using, 3D cell cultures, or sometimes we work with the organoids, tissue sections, whole brains. Even sometimes we started working with the model organisms like mouse or something. So there the challenges used to happen when we are working with the three-dimensional models. The major challenge is to capture the dynamics, because for capturing the dynamics, if we rely on a tool, means where the major challenge used to happen about the temporal resolution and all, and that's why we may miss sometimes we may miss the dynamics. And for catering such requirements, yes, different technologies we are using, where we mostly discuss about the resolution and all, multiplexing we discuss. And that's why Zeiss came up with the different solutions for you, where we have a different platforms for catering such applications, starting from the confocal platform. There are the different confocal platforms are also available that we called as light sheet. So yes, it is also a kind of imaging tool that we are using for the optical sectioning. So quickly, I'll jump not more about the technology but yes, more focused on the application part, that, okay, what are the challenges we are facing here? The idea for volume imaging, or if I say that, okay, idea for live cell imaging is that, okay, we are looking for a gentle imaging, we are looking for a long-term live cell imaging, we have to capture the faster dynamics, different combination of modalities we are looking for from one particular platform, so that we can do multiple things together. And yes, definitely one new tool that I have added here, that is instant volume imaging, so that you can capture the faster dynamics, which was not possible earlier. And now it is possible earlier with the new tool that Zeiss have introduced. So the first system that I want to highlight here, that is the Lightsheet 7. So nowadays, for getting the close relevance with the biology, we are working on the different biological samples or the 3D model. So it is a heartbeat that we are trying to capture here. So very fast acquisition. Yes. Here the thing that we are taking care of, lesser phototoxicity, better resolution, and yes, better temporal resolution also. So that you can capture faster dynamics with better resolution, or I can say that, okay, cellular and subcellular level you can achieve, and you can get the right data or meaningful data out of that. So the major challenge happened. What are the major challenges that we are facing on day-to-day life? So if I take the example of any EP fluorescence microscope, starting from your routine fluorescence microscope that you are using in your lab for capturing any type of sample, how it actually excite the sample, it will excite means when you have to capture a particular plane, you are simply illuminating the entire volume. So due to that, the long term, you can see here that, okay, it is highlighted in red. So means you are bleaching the other area except the area that you are trying to image. But with the help of light sheet imaging, so I'll say that, okay, only a plane or sheet of light that we are creating, and we are getting the focus signal only from this particular area. So the benefit here is that, okay, you are not bleaching the other area, and that's why you can do the very long-term live cell imaging. So if I take some example in terms of model organisms, so yes, long-term live cell imaging is possible. As you can see, the faster dynamics we are capturing as we are capturing the things into the volumes. And yes, it's not a live cell imaging for few hours. You can do the live cell imaging for a couple of days also. So that type of benefit is there. And yes, the overall idea is after capturing the data, if you have a tool for analyzing the data, I will say some meaningful data if you want to extract, then there are the different analysis softwares are available, which will help you to do the analysis not only in pixel by pixel mode. The smallest unit of the pixel is voxel by voxel. So we can do the analysis at voxel by voxel level, and that will give you the more close relevance with your biology. So another example, I think in case of plants, so we used to see the growth of the Arabidopsis flower. So that we are just trying to capture here. So again, for a couple of days, you just keep it there within the system and you can see the growth in real time. So yes, the new tool that I was talking about, the instant volume imaging tool, as you can see here, there is one tool that we have attached, and the system is equipped with the multi-array lens. So with the help of this multi-array lens, you can achieve the speed of 80 volumes per second, and that will help you to capture such faster dynamics. If I take the example of heartbeat or something, and those things are not possible with traditional point scanning confocal, or if I take the example of a spinning disk confocal, because there the speed that you are achieving, it is hardly 1 volume per second. But for capturing such faster dynamics, you require the speed of 80 volumes per second. So how it works actually, so if you have a sample here, so before the sample is being detected with the help of your objective lens, and the signal is projected onto your multi-lens array. And consider individual lenses as an individual eye. So suppose if you are checking one sample from by closing one eye, you can see the sample only from one angle. When you open your two eyes, you can see it from two angles. So similar fashion, if I have 37 different eyes, so I can check the same sample from 37 different angles. And that will help me out to get the parallax view. And yes, I can capture the sample from different angles. And ultimately, I will end up with the 3D result. And finally, when we process this data with the help of Zen software, we will get the entire 3D volume in single shot. And it will hardly take the time of 10 milliseconds, not more than that. So if I take the another view that, okay, how the sample will look like. So if you see your sample from one angle, how it looks like, from different angle, how it will look like. So ultimately, you are checking your sample from different angle. It will end up with your 3D result, and yes, process the data and you end up with your volume. So we'll end up with this type of volume. And yes, along why we called it as a 4D, because when you add the another dimension except X, Y, Z, the time factor when you add there, so it is your 4 dimension. So that's why we just name it as a light field 4D. So that will allow you to do the fast time-lapse experiment at 4-dimensional mode. Along with that, the major concern used to come that, okay, when we are doing the quantitative analysis, the field of view matters a lot. With traditional confocal system, the field of view is hardly 50 micron into 50 micron. But here the field of view is almost 10 times larger as compared to your routine point scanning confocal. So that will help you to do the quantitative analysis. Some other examples also that, okay, how the blood cells are flowing throughout the zebrafish embryo. So such type of events you can capture. Yes, as I said that, okay, there are the models are available. So if you have to do the work with the organoid or spheroid model, so such type of imaging is also possible, and you can simply count the number of cells. So such type of advanced analysis softwares are available now. So that will help you to do the advanced analysis along with your imaging. So another example down the line that, okay, if I have to check that, okay, how the blood cells are, insect blood cells are there within the Drosophila hemolymph, so how it can move. So those type of movements can be easily captured. So when we talk about the developmental biologist people, so they used to do this type of experiment quite often that, okay, the Drosophila movement, they used to check that, okay, how the signals are getting come out over the period of time. So if you just spin the Drosophila, then you cannot able to see the things in their natural state. So without tagging the animal, so you can simply do the things without any kind of special uh, special things there. So some other experiments also there that we have shown here that, okay, with Drosophila, okay, how the fat body movement is happening within the, when the pupa is forming. So such type of events you can simply capture. Another example again from Drosophila midgut cell lines been captured. So this example is very nice because most of the labs now they are working on the capturing the calcium signals. So if you have to see the calcium signal that, okay, how the calcium signals we can able to capture, because those there you require the very high temporal resolution. So there definitely the light field 4D is a very powerful tool if you have to capture any type of neuronal activity. As you can see here that, okay, we captured the neuronal activity at 10 volumes per second, as well as the field of view is quite large, which is approximately 9 times larger as compared to your traditional point scanning confocal system. You capture the dynamics and quickly you can jump to the another modality within the same platform that we called a super resolution mode for checking the morphological studies, if we have to do any kind of morphological study there. So side by side comparative analysis is possible, and that will give you more close relevance that, okay, whatever you are checking in terms of dynamics, that thing you can represent in terms of meaningful image also. So another quick example that, okay, how we can capture the overview. So within the system now, because yes, as we know that, okay, within the central facility, when we place any high-end setup, the major challenge used to happen with the new users. So focusing the sample, and that's why Zeiss came up with the new solution for the users that we called as artificial intelligence sample finder. So with the help of that, you can generate the map, and then wherever you want to capture the image with whatever the modalities, you can simply capture the images. So that type of flexibility will give you more with the system, and you can do more with the system, and you can generate more meaningful data. So quickly, I will jump to the another thing that whenever we talk about the organisms, so yes, we can talk about the cellular dynamics also. So when we talk, yes.
We have a time limit, so if you could just wrap up. I know it's a
Yes.
very exciting topic that you're
Just
discussing.
2 minutes. Just 2 minutes. In 2 minutes, I'll wrap up. Okay. So I just skipped this slide because this is also about the light sheet. But yes, in terms of application, what you can do. So some interesting thing, CAR T cell therapy, there also the microscopy plays a vital role if you have to do any targeting based thing. So there also it is possible. Another interesting sector is your stem cell colony imaging. So there also there is the powerful tool available from Zeiss. If I say that, okay, pulsating contraction, if you have to check, so such type of things are also possible. And yes, when it is, we talk about the organoid or spheroid model. So with point scanning confocal, the imaging is a bit challenging. So yes, the lattice light sheet is the solution there. With the help of that, you can capture the things from 5 different directions. So different models are there. So I'll quickly jump. So yes, Zeiss is providing a complete solution in terms of imaging. So that will help you to do more with the systems along with your different sample type. So thank you. Thanks a lot.
Thank you so much. And we are running short of time. So just one question.
One question can be taken.
Otherwise, we'll go to the next speaker. Thank you so much, Suparno.
Thank you, Mr. Suparno, for a very interesting presentation. Now I request the next speaker, Mr. Akshay, the Business Development Manager from Molecular Devices, and he will be speaking on the topic of advances in drug discovery.
presentation. Good afternoon, everyone. My name is Akshay. I'm from Molecular Devices. So today I'll take you through the drug screening in 3D. Like how we know, we usually perform cell cultures in 2D and everything. And when it comes to in clinical phase, we see many challenges. Some of the drugs won't pass through clinical phase because we don't know the complexity of the tissues or anything. So when we start doing like a drug treatment or anything in the organoid culture, we'll know the complexity of the cells and everything. So we'll have end-to-end details, how the morphology and where we can tackle, where we can target the drugs to the cells. Yeah, and to find out that, so we need a perfect organoid culture, or after that we need to, when it comes to screening, we need eye content imaging, or else if you're going for any output like luminescence based or anything, we need a good plate reader. Yeah, so I'll take you through one of the few examples like eye content imaging and 3D like automated 3D cell culture, where you can limit these applications. So Molecular Devices is a leading life science technology company that provides high performance bioanalytical measurement instruments, software, and reagents. And over the past few years, we have launched 30 plus new products with 110 patent innovations to support our technology. And this is our line of business, bioresearch, and plate eaters are our soul of our company, because we have sold n number of plate eaters. It may be single mode or multi mode, and reagents as well. And in drug discovery, you can see we have eye content imaging and flipper. If you want to see any real-time kinetics in calcium flux assays, or if you want to go for ERG assays to check the things, you can use go for flipper. And in biopharma, we have clone picks and Q picks for cell line and antibody development. So yeah, in traditional lab culture, you need to grow your organoid, and you need to feed them, or you need to treat them with a drug, and then you'll go for the analysis through the imager, or like luminescence based or fluorescence based. And instead of that, we have automated cell culture unit, so which can perform everything for you, and you can direct it to either for imaging, or you can go for either other outputs. So this is a Cell Express automated cell culture. Usually, if you're working in a CRO or anywhere, so we spend around 2 to 3 hours for cell culture maintenance or anything. Like daily, we need to maintain cell culture, and we need to see the growth. And mainly, if you come to organoid culture, the contamination is the main thing. So with this one, there is no issue of the contamination, and there is no like tracking the organoid or anything. So once you feed the system, so what are your cycles and everything, it can take up. So this is a liquid handler. This is the liquid handler, where it has stage with robotic arms, where you can store the plates, and you can program it. Either you can go for drug or feeding of media or anything like that. And we have incubator, which can store up to 96 plates together. And we have imager inbuilt in the system, where you can time to time you can check your organoid, how it's growing, and what is its functionality. And we have a compartment of media storage, where it can hold up to 2 liters of 2 bottles. And we have touch screen, so you can enable the program, and you can carry on. So this is the stage. We have it can store 5096 plate at a time, and we have 8-span pipette. It's a robotic. You can go for compound dilution or anything like that.
And we have a deck position, like it has heating and cooling mechanism with tilting. So if you want to change the media or anything, you will not able to disturb the cells. So you can use that. So this is how the plate elevator works. Now the plate elevator is in the stage. So you can, so the system will perform if you want to media change or anything. After it is done, it will directly go back to the incubator stacker. So where the plates are stored. So it is maintained completely with sterility. And we have a camera also. Yeah, and we have HEPA filters and UV sterilization to maintain sterility. And this is a camera which is inbuilt. And this is a software. It's a very user-intuitive software. In the 5 protocol steps, you can set your protocol, like either be it seeding, feeding, or incubating. And you can set your protocol like it can go up to like n number of days. So if you are if you want to seed today and after the organoid is developed, if you want to treat the organoid with the drug, you can program it and you can see it. And this is the how the organoid growth you can measure with the visualization. You can track the stem cell growth and everything. And now the organoid is grown, and now we want to visualize in eye content perform. So in that you can see there is only one like lower version of the camera. So in the Image Express SCS AI, it is an advanced camera, and this can go up to from 612 plate to 1536 plates. So we have 3 configuration in this. One comes with wide field, confocal and advanced. Wide field comes with LEDs. Confocal comes with a dual spinning disc and LED. Advanced comes with dual spinning disc with 7 lasers. So this can be free upgradable. So this is the objective. It can be placed 6 objective at a time, and the magnification ranges from 2x to 90x. So one objective can perform 2 magnification because we have one special component called magnification changer. So it can magnify if you want to image, if you've taken an image in 2x, if you change that into magnification changer like 1.5, it can go up to like 3.5x. So in the 60x, you can view 60x and 90x together. And we have confocal disc. We have pinhole and slit confocals for the deep tissue or any 3D imaging. And this is a camera. It's a high sensitive CMOS camera. And we have environmental control, which comes with the CO2 gas and temperature control, and you can perform some hypoxia experiments also. And if you want to see the kinetics growth like 7 days or 7 days or more than that, you can go for it. So it can maintain sterility and also the environment required by the cells. And this is a software, and this is a very useful, like very less complicated software or very intuitive software. If you see, there are only 5 steps. In the first step, says select your plate and configure the channels, like if you want to go for wide field or confocal, or if you want to go for 20x or 30x or anything like that. And in the next step, you can select the sites to if you want to cover the entire well or you want to cover only one particular type of well. So that, and channels, you can select all the 7 channels together, and yeah, you can select the channels and you can set the exposure here if it's, and we have autofocus and software based autofocus and hardware based autofocus to measure the well depth and the sample also. And yeah, now we are ready to run the plate. So this is the MetaXpert software. So this is built with AI in-kata analysis software, where it can, if you set the protocol like I want to see the organoid, and after that, in like if you keep the next plate, it will only target the cells. So it has quick ID, which can target the cells and it can tell you the area, volume, and everything while the screening is happening. So this is an in-kata software where you can see all the analysis present here. If you want to go for volume, measurement of the nuclei or puncta or network fibers, everything. So these are the some example images, and these are applications. And if you are going for tumoroid assay flow, so you can culture the organoid in the automated cell culture, and you can go for imaging with HCSAI. And second application is iPSC derived 3D hepatocytes toxicity study. And these are some examples for if you're for studying for neurotoxicity that can be screened and done. And this is the intestinal organoid workflow. Yeah, thank you. If you have any questions, you can ask.
Thank you, Mr. Akshay, for very nice presentation.
Question.
Yeah.
I mean, having such an instrument, I mean, we don't really need humans to do anything anymore. I mean, you're creating an organoid and you're analyzing the organoid. You don't have to have the experience in cell culture to actually do this kind of work, right?
Basic knowledge is enough. Like
Just theoretical knowledge.
theoretical
no practical
knowledge
required.
practical and is if the person has, it's very good because if he knows to interpret how the organoid is growing, that knowledge should be there. Or practically, it can manage, the instrument can manage.
Thank you so much.
Yeah.
Thank you.
Thank you.
Now I request the next speaker of the session, Dr. Sumir Rena. He's a commercial marketing manager from Merck. Will be speaking on the title Elevating Neurobiology Research.
Okay, thank you. A very good afternoon to all of you. I think I am the last presenter, and I know I am between you and your lunch, so please bear with me if you are feeling hungry. So I'm Sumir. I represent Merck here, and I am a marketing manager with Merck. I take care of the protein workflow portfolio for Merck. I did my PhD from Actrac Tata Memorial Center. I was like, you know, most of you doing their research work, and then I shifted to the other side. At Merck, you know, our basic objective is to kind of provide you innovative products and tools so that the research you are doing currently in your lab, you know, can be done very efficiently, reproducibly, and you know, every time you do, you get the same kind of result. So that is the objective with which our R&D team works at the back end. The title today, I will be speaking about you know, how the innovations in Western blotting and electrophoresis have revolutionized. You will see that, you know, how quickly and reproducibly you can do a lot of your electrophoresis and Western blotting experiments. One of the speakers earlier told about, you know, how the PCR has evolved over the years. To give you a background of it, maybe some of you would know that this is how PCR used to be done earlier. You know, in late 60s, 70s, there were these water baths where you would do your denaturation, your annealing, your extension by, you know, physically taking out the tubes, putting it into the water bath, and then, you know, doing these 3 or 4 different steps of the PCR. And look at how the technology has evolved, and now you have these small benchtop instruments where you just put your tube, you put DNTPs, master mix, your DNA sample, and that's it. That's how quickly the PCR can be done now. Our colleague from Bio-Rad also showed you, you know, how different, you know, how many different versions of the PCR going up to the digital PCR or the droplet PCR has come up now. And now if you look at Western blotting, this is just a trivia I wanted to share with you. You just do a simple PubMed search of Western blot, and you will get around over 4 lakh citations or publications which have done this experiment, which tells you how critical or important this technology is still for protein detection and semi-quantitation. So that is where our team thought that let us understand what are the challenges researchers like you face in the lab whenever you are doing an electrophoresis or a Western blotting experiment, and how Merck can help you in overcoming those challenges. So with that in mind, this is just a snapshot of what all we have in our portfolio, the complete portfolio. If you are doing a Western blotting gel electrophoresis experiment, Merck is one point of source for consumable instruments and any other product you would be requiring for your experiments. But today I will be talking about just spend 2 minutes on the two major challenges which our electrophoresis Western blotting researchers kind of face. One is leakage. Every time you are making your gel, you find leakage. With the raise of hand, any one of you have faced this challenge, that whenever you are pouring your resolving stacking gel, right? Few of you have faced this challenge, right? So this is something which you do not want. You are setting up your experiment, and then you see you have made dissolving, you have made stacking gel, you pour it, and it gets leak, and you have to repeat the experiment. Second thing, breakage of your plates, your short plates and your long plates. Again, raise of hand, how many of you have faced that challenge also, right? Few of you, exactly. So that is what our R&D team thought, that what we can do to kind of overcome this challenge, and that is where we introduced a small design change in our casters. Now these casters, we say these are 100% leak proof. Why? Because we have introduced a lock which takes care of the alignment of the long plate and the short plate, and also the locking mechanism has been changed so that it does not directly interact with your plates, thereby taking care that the plates are not broken. So these are for certain small changes we have done, very important, because it takes care of these two critical problems which a lot of our researchers currently still face. And also I don't know, some of you might have faced this challenge, that you set up the experiment, load your samples, you switch on the current and voltage, you go for lunch, you come back, and then you see that the bands have not moved at all. That is because maybe the electrodes were not in the proper orientation. What we have done here is we have designed the cap or the lid in a way that it will fit only if both the electrodes are in the proper orientation. So you will say these are like very small changes or very small design changes, but you can see how effective they are in terms of how your overall experiment is performing now. And second thing is for gel casting, most of you would be making your resolving solution, stacking solution. Few of you, if the budget allows, you switch to a precast gel, which is a ready-made gel. But what is the price difference between when you do it in your lab, a home brew method, and when you switch to a precast gel? It is almost 8 to 10 times of money you have to spend for just one gel. And that is again one feedback we received from a lot of our researchers. Our R&D team sat back and thought, what we can do about it. And this is what we have come up with. This is called as Ampage Lux Gel Casting System. The first half of the image here is what you are currently doing in your lab. You make resolving solutions, you pour it, wait for it to polymerize, and then you make a stacking solution. The whole process takes around 30 minutes, 45 minutes, in some cases 1 hour to just make the gel. That is just the preparatory phase. Now you load your samples, another 1 or 2 hours. The whole experiment becomes very, very long. Now, what we have done here, we have introduced this technology, Ampage Lux, where you would see that now, instead of spending 90 minutes, the whole thing can be done in less than 3 minutes. You're resolving, stacking, polymerization, everything in under 3 minutes. And why? How can we do that? The technology is a UV-based technology. We have this instrument, which is called as Ampage Lux. We provide reagents which have photoinitiators in it. So when they come in contact with the UV light, the polymerization is very, very fast. Polymerization, the only polymerization step only requires 90 seconds. So you can imagine how fast or how quickly you can make your gels. And very simple to use. You put on your caster inside this machine. You pour your resolving. You pour your stacking also at the same time. You do not wait. Earlier, you wait for polymerization to resolving to polymerize first and then the stacking step. So that is gone. That is point number one. There is no APS, no timid requirement. So now we are shifting to greener chemistry also. And that is a mandate. I'm sure a lot of researchers also now, they want to shift to greener technologies. That is what we have also incorporated here. Does not matter if you are working, if you are doing a semi-dry method or a wet transfer or a turbo system, any transfer method you use after running the gel, or these gels are compatible with that. It does not matter if you are running 1, 2, or 3, or you are running your experiments in triplicate. That batch-to-batch variation is gone, because it's very easy to make the reagents now and to make the gel highly reproducible every time. And it does not matter if you are making the gel today, 12 months down the line, or 24 months after you have purchased this reagent, because the shelf life of the product is 2 years. So, these with these benefits, and I initially talked about, from a hand casting, if you have to switch to a precast gels, the cost goes up. You do not want that, because if you are spending, say, for example, 100 to 200 rupees for preparing one gel, if you switch to a precast gel, the price goes to 800 or 900 rupees. If you switch to this technology, the price goes up only by approximately 50 rupees. So now you are spending, instead of 200, you're spending 250 or 260 rupees per gel. So the economics of that is also what we have taken care of, apart from the innovation we have introduced into this technology. So this is the overall portfolio we offer. As I said at the beginning of my presentation, that if you are working if you are doing a electrophoresis Western blotting experiment, we provide end-to-end solutions. What does it mean? We provide all the consumables, we provide instruments, power supplies, and few of the innovative products. So I just spoke about to you. I'm sure all of you know that Merck is an umbrella company. We have Sigma, we have Millipore brands also with us. And I'm sure, I strongly believe that most of you would have used Merck products, in somewhere down the line during their experiments. We have more than 3 lakh products in our portfolio. But these are, some innovations I wanted to just show to you. We are currently running an offer on these, which is till March end. So if you are looking for any of these products, it can be membranes, it can be HRP substrate, or any other instruments for electrophoresis or transfer, we are providing these things to you. Thank you very much. I hope I was on time. Thanks a lot.
Any questions? One quick question.
Minutes, the casting technology is really good. So I have a question, like, suppose if someone has the BioRad setup, the BioRad glass setup and everything, can we still use the 3-minute solution to cast the gel in it?
So one thing is, first part, we provide a caster along with it. So it does not matter what electrophoresis unit you have. For example, you already have a lot of BioRad material with you. This is perfectly compatible with it. Even it is compatible with Invitrogen and Cytiva, casters and electrophoresis units. So
Okay.
that way it is quite open.
Okay. And my second question is, will the BioRad casting setup will go inside the UV?
BioRad has a two gel casting setup.
Right,
So
right.
this is a single caster.
Single, so it
and
won't.
we provide 2 casters along with the instrument. When you buy, you get a 2 casters, you get 10 plates, combs. The complete set is provided to you.
Okay, okay. Thank you so much.
Yes, sure.
Apart from APS and TEMET, rest everything can be used along with the photo initializer, or the rest things have to be also changed?
Yeah, so the resolving solution as well as the stacking solution we will be providing. We provide a kit.
The entire thing should
Yes,
be taken.
yes. And as I said, it does not require a TIMID or a APS to do it.
That was obvious.
So you just pour the reagent, you put it into the instrument, switch on it for 90 seconds, your gel is polymerized.
But then the entire thing has to be in the same club.
Yes, yeah, right.
Thank you.
Any other questions? Okay, thank you so much, Mr. Rana.
Thank you very much.
Thank you. Our last speaker is Mr. Sridhar Tiwari. He's the application manager at Leica, and he will be talking on laser microdissection, opening frontiers to molecular biology research.
So, very good afternoon, everyone, and thanks to the organizers for giving us the opportunity to present our product. So, I think Akhil has already set up the context, why we need this kind of system, which is a kind of, I would say, workflow based system. Okay, one of the first Leica's workflow-based systems, and why we need such products. We do a lot of risk marker-based studies where we want to study the different maybe genomic markers, or maybe into proteomics, or maybe some metabolites, out of a disease sample, maybe a cancer tissue, or any other human sample, right? But at the end of the day, what we isolate is a kind of cocktail which can include even both your infected, for example, DNA, which can include a cocktail of both normal as well as the cancer cells DNA. Right. So this is one technique which can help you in very specifically isolating the infected versus normal cells, and then making your study very specific to the the infected cells DNAs or the cancer cell DNAs. So why we do laser microdissection is because we want to isolate those single cells or the features which we want to analyze out of the the non-homogeneous population of the cells. Okay. So what exactly is laser microdissection? So this is a technique where we use a UV laser and we try to isolate single cells, or maybe some cells with specific features out of the heterogeneous population of cells from a tissue section, or it can be any other sample. Okay. So, I mean, it is with Leica, it is more like purification under visual control. We want to see what exactly we are isolating, and at the same time, we want to, segregate the different type of cell populations. Okay. So the key benefits what we get with the laser microdissection based systems is we want to isolate the cells, and it should be contamination free. We don't want to mix up the infected versus non-infected cells, and at the same time we want to see what exactly we are cutting down and where exactly it is going. Okay, so the basic principle how it works. Obviously, you need a UV laser which can hit your sample, which can be precisely focused on the features or the cells which you want to dissect. And also, you need certain optics, and you want to place your sample in a membrane, and the laser hits your sample through the optics, and it slices down your desired features, and it gets collected to your PCR tubes or maybe multi-well plates, and which is followed by your downstream analysis, which can be either genomic analysis, maybe you want to do NGS, or you want to do mass spec, or maybe some metabolite analysis. Okay. So for this, there are different type of consumables which are required. Consumables, mainly these are membrane-based slides, or it can be petri dishes for even the live cell samples, okay, over which we place our sample, and so that it can be dissected easily. Now,
what exactly it includes, already I have explained a lot. So these are some of the main applications where we can use the laser micro dissection systems. Most commonly used ones like cancer biology, pathology, molecular research. Almost every field you can use a laser micro dissection system. Even a lot of plant biologists, they use laser micro dissection systems to isolate the desired features or structures out of the whole sample. Okay. These are some of the common applications, but it is not limited to only these applications, which can include right from genomics to proteomics to metabolomics, or even the plant research. You want to isolate even the individual chromosomes. That can be done using laser micro dissections systems. And the latest one where we want to dissect the live cell samples. That is also possible using the laser micro dissection systems. And I mean, the latest concept, which is now getting very popular, the deep visual proteomics. That's the new concept which has emerged in the recent time. That's another application which can be done on the laser micro dissection systems. Okay. So, to prepare the sample, as I said, you need a sample carrier. It can be a slide, it can be a petri dish, or even the multiple plates, which has a membrane in the bottom over which your sample is placed and dissected along with the membrane. Okay. And then, let's talk about the Leica laser micro dissection system. Technically, this was our first workflow based system. I mean, it's a part of the workflow. You can do imaging as well because it is based on a microscope. So apart from imaging, you can isolate your desired features or the cells by dissecting it through the laser. So it's a part of workflow. Can be your genomics workflow or proteomics or metabolomics workflow. Okay. So talking about the hardware, basically you need the laser which can dissect. So Leica gives you both the options for just the routine purpose. We have 70 microjoules laser. So if somebody is looking for high throughput kind of applications where they have huge sample load or maybe harder samples, you can go for a higher power laser, that is 120 microjoules laser. And then definitely you have the microscope. I don't know whether this is working on. Yeah. So okay, I'm sorry, this is not working. So I mean, that's the microscope. We have a system which is based on an upright microscope, unlike the conventional concept, which was based on the inverted microscopes. And then, depending on your applications or maybe your likings, you can have different type of stages. Okay, so how exactly it works? With Leica, it is more like, your laser is precisely focused on your sample because the movement is via optics, unlike the conventional concept where, your laser spot is fixed and you are moving the stage to align it as per your, laser spot. So what Leica has done is we have integrated our laser in a way that your laser spot is moving through the optics, which makes it more precise and less frequent alignments required. And this also helps you in real-time cutting of your cells. Okay. Now this the laser-based cell capture or the dissection, it's not limited to just the bright field samples, because nowadays we are more interested in studying individual maybe proteins or structures through fluorescence, which is more specific technique. So so that's where these laser micro dissection systems, they extend your capabilities to fluorescence-based dissection as well. So for that, we provide special filter cubes which are compatible with the lasers. And obviously, this is something which user has to let us know that whether they
want to do the dissection in fluorescence mode or not. But yeah, I mean, this is further extending your dissection capabilities to fluorescence mode as well. Okay. And because of this upright microscope, like I said initially, that we have a different kind of design, which is upright based, which is more familiar kind of microscope system, because this is something which we have worked on a daily basis. And added benefit when it comes to dissection is the collection of the sample is with gravity. Unlike the conventional catapult based, where you are pushing the things against the gravity and there's a lot of sample loss. So once you are collecting the sample, dissecting and collecting it with gravity, there is lesser sample loss, and there is minimum chances of cross contamination. Okay. So couple of applications where it can be used. It can be used for genomic studies. This is a typical DNA workflow, or it can be used for even the RNA analysis also. I'll take maybe just 2 minutes more. Proteomics, this is something which is getting a lot of attention nowadays, especially with the deep visual proteomics. I'll come to that maybe in the next slide. And this is another application where people are looking for the laser-based dissection systems a lot, working with the live cells. Now we are offering a upright microscope, but this is not limiting your work to the tissue sections only, means the samples on a slide. You can do a live cell cutting as well, despite using an upright microscope. This is because this is a specially designed microscope which can accommodate even the Petri dishes as well. Okay. So this is the the new normal, because see, nowadays the marker-based studies we are doing a lot, and it is not like the CROs or pharma companies, they may not be working with maybe 1 or 2 samples in a day, maybe 10, 50 or 100 of samples in a day. So where I want to increase my throughput for cutting for dissection. So this is another technique which has emerged in recent time, where you can capture an image. We do AI-based segmentation. You classify the different features which you have in your sample, and along with the coordinates, this whole classification can be imported to your laser microdissection system, and after that, your LMD system can automatically detect these different features and dissect them. Not only dissecting, but it can also push it to individual tubes, PCR tubes, which again you can assign, which makes your job a little easy, right? This is a simple, I tried to explain the DVP in a simplest format. If somebody is interested to know more details of DVP, I'll be happy to explain to you. Maybe we can connect after the session. Okay, so that was all from my side. If you have any questions, I'll be happy to answer that, or else post the session, we can connect and discuss.
Thank you, Mr. Sridhar, for a very interesting talk. Any question? Any query? Okay, thank you very much. And
thank you, sir.
Just to conclude this session, I sincerely like to thank all the 7 speakers of the session for very informative presentations. And it is really very heartening to know the integration of latest disruptive technology in the existing technology as well as traditional equipment to make them more user-friendly, more result-oriented, and more precise, and which is in line with the Industry 5.0. So I once again thanks all the speakers. Thank you.
With this enriching session, we conclude the final technical segment of the conference. On the behalf of the ICoMB 26 Organizing Committee, we would like to express our sincere thanks to all the speakers from the industry for sharing their valuable, informative talks. We request our respected chairperson and co-chairperson to kindly present mementos to the distinguished speakers as a token of appreciation for their contributions.
May I request Ms. Shobha Iyer to please come on stage, and requesting the chairperson and co-chairperson to please give them awards. Ms. Shobha Iyer? She's not here. Mr. Akhil Gupta from Biorad Laboratories. Please put your hands together to May I now request Dr. Surbhi Gupta from Horiba Industry India Limited. Please give her a huge round of applause. May I request Mr. Akshay from Molecular Devices to please come forward? Mr. Akshay. Okay. So may I request Dr. Sumir Raina from Merck to please come on stage? May I request Mr. Sridhar Tiwari from Leica to please come on stage? Please give him a huge round of applause.
We would also like to take the opportunity to honor our esteemed chairperson and co-chairperson for their guidance and support throughout the session.
May we request Dr. V. Pooja, Director AIB.
Kindly honor the chairperson and co-chairperson, please.
Also requesting Dr. Puniti Mathur to please come on stage to do the honors. So please put your hands together to thank their efforts.
Once again, we thank everyone for making this session a success. Sorry. Once again, we thank everyone for making this session a success. And now we are proceeding for the lunch break. We request all the registered delegates and the students to kindly proceed to F2 foyer for the lunch. And all other faculties will have at F1 lounge. All our invited distinguished speakers will have their lunch at I2 lounge.
So you're also requested to come back to the same place as we will have our valedictory, which is scheduled from 3 onwards. Thank you.
So now, Rishika, वो अकेले थोड़ी बैठेंगे अरे बैठ जाएंगे तुम चिंता मत करो सेटिंग तो नहीं है ना मैं May I request Dr. Selvamurthy, Dr. Vijayan, Dr. Amitabha to please come on the stage, please.
A very good afternoon to respected dignitaries on the dais, eminent speakers, delegates, faculty members, participants, and students. On behalf of Honorable Founder President, Dr. Ashok K. Chauhan, Honorable Chancellor, Dr. Atul Chauhan, Honorable Vice Chancellor, Dr. Balwinder Shukla, and Organizing Committee, it gives me immense pleasure to welcome you all to the valedictory session of the International Conference on Medical Biotechnology, ICoMB 2026. We are deeply honored by the august presence of Dr. W. Selvamurthy, President, Amity Science, Technology and Innovation Foundation, Amity University, Professor Dr. V. Pooja, Director, Amity Institute of Biotechnology and Chairperson, ICoMB 2026, Dr. Edithil Vijayan, President, SBTI, Professor Dr. Sudeep Bose, Head, Center for Medical Biotechnology, AIB, and Co-Chairperson, ICoMB 2026, Professor Dr. Amitabh Mukhopadhyay, Co-Chairperson, ICoMB 2026, Dr. Banashri Bandyopadhyay, Organizing Secretary, ICoMB 2026, and recently elected on Executive Council Member, SBTI. We are waiting the arrival of our Vice Chancellor, Professor Dr. Balwinder Shukla, who will be soon joining us.
Over the past 3 days, this conference has witnessed stimulating scientific deliberations, interdisciplinary discussions, and meaningful interactions that have significantly contributed to the advancement of medical biotechnology. As we reach the conclusion of this enriching academic journey, today's session brings an opportunity to reflect upon the outcome and celebrate excellence.
To formally begin the valedictory session, it is my privilege to invite Professor Dr. Pooja Vijayaraghavan, Chairperson of the conference and Director, Amity Institute of Biotechnology, to deliver the welcome address. Over to you, ma'am.
Thank you so much. I would request the students to kindly settle down. Thank you so much. On behalf of Honorable Founder President, Dr. Ashok Chauhan, our Honorable Chancellor, Dr. Atul Chauhan, Honorable Vice Chancellor, Dr. Balwinder Shukla, our respected Dr. Selvamurthy, Dr. Vijayan, and everybody who's on the dais and off the dais, our eminent speakers,
my colleagues, researchers, young scientists, and my dear students. After 3 days of excitement, today we come to the concluding moments of ICoMB 2026. And it gives me immense pleasure and a very deep sense of satisfaction to reflect upon the remarkable scientific journey that we have collectively experienced over these past 3 days. We started on 6th, and the entire report would be presented by Dr. Sudeep Bose, but the conference, which was held from 7th to 9th of January, provided platform which was truly vibrant in all senses. It was a confluence of ideas, innovations, translational science, and global collaboration. These 3 days witnessed involvement of scientists who are the best in our country. Starting with the inaugural session, where Dr. Shekhar Mande and Dr. Madhu Dikshit was here. Their talks were not only intellectually enriching, but were also deeply motivating for researchers across generations. Over the course of 3 days, we witnessed not only internationally renowned scientists, but also directors and Bhatnagar awardees from across the country. Dr. Deepak Nair, Dr. Sanjeev Khosla, Dr. Dulal Panda, Dr. Bushra Atik, Dr. Vinay Nandikuri, and many others were here to deliver their talk, their cutting-edge research, and the advances that they have done in the course of their scientific journey. We deliberated on so many scientific topics, right from molecular mechanism of diseases, cancer biology, cell signaling, molecular pathogenesis, immunotherapy, genomics, neuroscience. I can go on and on. There were 3 parallel sessions, and it was an overwhelming response with all the sessions chock-a-blocked with students and researchers. It was particularly heartening was the active engagement of all of you. From early morning keynote sessions to the late night, we witnessed students, faculty members, and everybody present in the hall, which itself shows how enriching these 3 days have been. This is just the start. ICoMB 2026 is a beginning. It is not the end. Very soon, all of you would be getting invitations for ICoMB 2027, which we would start with the blessings of Honorable Founder President, Sir. I believe that this beginning will reap great benefits because we have seen the discussions which happened over these 3 days of involvement, the support and the guidance promised by all our directors and senior members who came and witnessed, and the overwhelming response from the scientific community. The outcome of these 3 days would be extremely rewarding for all of us. And with this thought and with this belief, I once again thank all of you for being a part of ICoMB 2026. And I eagerly look forward to meeting you all again in ICoMB 2027. Thank you so much.
Thank you, ma'am. It's our honor to introduce Dr. W. Silvamurthy, President of the Amity Science, Technology and Innovation Foundation, Director General of the Amity Directorate of Science and Innovation, and Chancellor of Amity University, Chhattisgarh. Dr. Silvamurthy has been instrumental in augmenting the research activities at Amity University. A distinguished scientist and former Chief Controller at DRDO, he served the Ministry of Defense for nearly 4 decades, leading life sciences and biomedical research for the armed forces. In recognition for his significant contributions in the field of biomedical research and development, he has been awarded several prestigious research awards. A few prominent ones are Yoga Ratna Award by Shwasa Yoga Center, Dr. APJ Abul Kalam Award for Most Prominent Scientist and Educationist by Oasis World Records, CSIR National Award for Science and Technology Innovations presented by the Honorable Prime Minister of India, and multiple Lifetime Achievement Awards for scientific leadership. I respectfully request Dr. W. Silvamurthy to address the August gathering.
It's always clapping is good for your health. be happy always. This is what we are created for. I would also like to be always happy, and I want to reflect, radiate happiness around. So let us celebrate our life. We are alive. We should be happy, and we are fit. We are healthy. Another happiness. And we are blessed with so many things in life. So let's be happy. So clapping is good. Let's clap once again. Great. So good afternoon to every one of you. And I'm so happy to be here at this valedictory session of the 3 days very valuable conference which you have heard, and the Amity has organized in a big way with the blessings from Honorable Founder President, Dr. Ashok Chauhanji, and also the Chancellor, Dr. Atul Chauhan, the help of Vice Chancellor, Dr. Balvinder Shukla, with the great leadership of Dr. V. Pooja, the Director of Amity Institute of Biotechnology, with all the help and cooperation from every one of those staff here in biotechnology. I must make special mention about Amitabh Mukhopadhyaya, who is one of our star scientists in the Amity Institute of Biotechnology. We are privileged to have such a great leader here, and who has done research in terms of biotechnology projects, patents, publications, a well-known figure who was responsible for the success of this event. Let's give a big clap to Dr. Amitabh Mukhopadhyaya, along with the whole team of Amity Institute of Biotechnology. I must mention a few names in this: Dr. Chandradeep Tendon, Dr. Simran Tendon. Then we have also Dr. Sandeep Bose, who is sitting there. And also we have Banashri, Dr. Banashri, and many more. I have just mentioned only a few names, so they all deserve a big round of applause. Let's give a big round of applause to all of them. I must place on record our gratitude to the president of the society, the Biotechnology Society of India, which I was told 5,000 membership. It's a huge society. It's almost, the life membership is about 1,300. And the total number, 3,000 and above, is the other general membership. Put together, 5,000 is a big force. So I'm sure this society, which Dr. Vijayan is the president, I'm sure it will bring out many, many new innovations, facilitate such kind of interactions, forums, create consortium of industry and the scientists and faculties. So you have a big responsibility, Dr. Vijayan. We are all there with you. Because I know Dr. Vijayan since 70s, because since my DRDO days, when he was in Kochi in the biotechnology department there. And so I have a long association with you, the president of the society. So we are also privileged to host this event, annual conference. So we are so grateful to you, Dr. Vijayan, and the whole society, and Dr. Rao from NIMHANS. So I have interacted with all of them. So because NIMHANS, I used to go there as a trainee there. I have been there in 70s when Dr. Raju was there. I used to visit there and learn EEG, recording of EEG, stereotaxy, and also microelectrode recording, patch clamp for the ionic studies, all that. So he is from that lab. So I also know Dr. Rao for quite some time. So all these members and the president is taking the society for biotechnology to a newer pinnacle, because the 21st century belongs to life sciences. If you look at 20th century engineering and 19th century physical sciences, but 21st century belongs to you, belongs to biotechnology, belongs to biosciences, life sciences. So this is a century of life sciences. So a lot of exciting things will happen in the field of biotechnology. So I'm sure all of you, particularly my young scholars, the future leaders in biotechnology who are sitting at the back, and this is a message for you, the opportunities are plenty. If you look at the global economy today for biotechnology, and it is projected to be, currently it is $1.8 trillion. Imagine $1.8 trillion, almost $2 trillion is the global market, which is rising at almost 15%, 14 to 15% as the CAGR So every annual growth rate for this world market is going to be huge, and it is going to reach about $5.8 trillion in the next 10 years. By about 20, I have just taken the statistics. So in the next 10 years, it is going to reach about $6 trillion. So this is the opportunity for you. Bioeconomy is growing in such a big way. When you look at India, this is global. When you look at India, it is about 170 billion. So trillion, billion. We are in 170 billion. And then it is expected to grow in the next 5 years into 300 trillion. So that is the growth rate. So youngsters and also our faculties, this is a great opportunity for all of you to look at plant, animal, human, medical biotechnology. All this you can work together and then come out with many things. In bioindustries is one, biopharma is another area, and there are many, many bioprocessing, everything. There's a great opportunity for you to work. And I appreciate the biotechnology department, which is a huge department, which Dr. Pooja heads. Earlier, Dr. Chandradeep Tandon used to be the director of this prestigious, the Amity Institute of Biotechnology. And this is a star institute for us because we have huge number of faculties, almost more than 100, and we have 50 projects, ongoing projects, and of the value of several crores. I don't want to mention the figure actually. And then this, so number of PhD scholars, number of the technologies coming up from this in all the plant, animal, and human biotechnology. And I'm so happy that they have taken the initiative to organize this. Now, coming to the area which I want to mention, highlight, what are the priorities which the country should look at? One is the gene therapy and cell therapy. This is going to be a great opportunity. In biotechnology, gene therapy is now growing very fast, and you can edit genes, gene editing. You have so many now tools available to edit genes, the defective genes, remove and then put the right gene, stitch it, and then ultimately solve the problems, whether it is the lack of protein synthesis. So you can do all this type of biotechnology manipulation. So gene therapy and cell therapy is one field. Synthetic biology is yet another field, synthetic biology. This has got both the biodefense implications as well as the medical therapeutic applications, synthetic biology. Then field-based diagnostics, because we need quickly, because you don't have to bring the samples here. If you are able to diagnose the patient right in the field, it's so helpful. So you need field-based diagnostics for large-scale screening, cost-effective diagnostics, yet another very important field. Then vaccines is very huge. We are already the global hub of vaccines is India. Bharat Biotech, Shanta Biotech, we have Biocon, so many industries are coming in the field of vaccines and including Reddy's laboratory. So we have close collaboration with all of them. So vaccines is going to be yet another area for many diseases. Now, including cancer, we are looking at, can we have vaccines, like vaccine for cancer cervix, which is HPV, human papilloma virus. How do you treat them? Can we vaccinate? Now it's being applied. So like this, vaccines is a very huge field, but then AI applications in vaccine development, we should look at that. Amity has done some very interesting work in artificial intelligence application, because if you have, say, about 50 candidates, potential candidate for vaccine, how do you downstream to 2 or 3 potential successful vaccines? So this, you can use it by data analytics as well as the artificial intelligence. So use AI tools in such new drug discovery, as well as in the new medical drug discovery, as well as vaccine development. The other area is the biologics as well as the biosimilars. This is again another potential area, biomimetics and biosimilars. Then precision and personalized medicine. This is also a very important area, pharmacogenomics. That how do you give a customized precision medicine? Let's welcome Honorable Vice Chancellor, Dr. Balvinder Shukla ji. How are you? Let's welcome our Pro Vice Chancellor, Dr. Chandradeep Tandon, whom I mentioned just a few minutes back. And he is the one who headed this biotechnology. He himself is a biotechnologist. And now Pooja has taken over from him. So we welcome both our Vice Chancellor, Dr. Balvinder Shukla, as well as the Pro Vice Chancellor, Dr. Chandradeep Tandon, amidst us. So the precision and personalized medicine could be another area using pharmacogenomics. Coming to the final carry-home message I want to give in my address, that opportunities are plenty, but how do we harness these opportunities? What do we do with our competence, capabilities as a group? So that is where this type of conference, which Dr. Vijayan has given to Amity, this organization has given to us, we must harness this potential of connectivity, networking, collaborations, what you have brought. He is from NIMHANS, he is from DIPAS, he is from DRDO, so they are from SRM. So you have so many people coming. How do we network together as one single force? So the consortium approach, collaborative approach, synergy approach is one message I want to give. We are excellent as individuals, small groups, but we have to work as a large team. This is what happens in developed world. So you do things faster, do things better. So this type of societies, like biotechnology society, gives opportunity for us to connect with each other. So connect with each other. This is what founder president Dr. Chauhan has given 3 missions. Mission connect, mission synergy, and mission outcome. So this should bring a very fruitful outcome. In fact, Dr. Ashok Chauhan, founder president, has sent a message to Dr. Pooja today morning, create an outcome committee. And this outcome committee should now give a roadmap. What are we going to do in the field of biotechnology, which she has already discussed with me, that we will now make a roadmap based on the deliberations, the priority areas identified, and the collaborations built with collaborators from institutions, universities, and also industry. What do we do from here? So what should be the outcome? Outcome for Amity, outcome for you as participants, and outcome for the country and humanity. So this is what is the outcome. So we will be looking forward to Pooja, that outcome from these deliberations. And I'm sure that you will bring out a very valuable roadmap and clear goals. And let us contribute to bioeconomy, because the growth rate, as I mentioned, is 15% CAGR every year and reaching to $5 trillion market. So how do we contribute to Indian contribution to the global market? We should increase that. So let us look at how do we translate our leads, scientific leads, into a product, technology, or a process, giving the deliverable to the society. So this is what we should now look at. Finally, we must all contribute to the Viksit Bharat. This is the vision the Honorable Prime Minister has given to our country, developed country by 2047, in which biotechnology will contribute to our bioeconomy, our GDP. GDP currently, our contribution is 8%, but then we should increase biotechnology contribution. So we must enhance it because there's a potential is very huge, and I'm sure it will happen. And I wish, I'm so happy that Amity Institute of Biotechnology, with the blessings of our founder president, Chancellor Dr. Atul Chauhan, and also Vice Chancellor Dr. Balvinder Shukla, have been able to organize this. And I hope that you had a wonderful interaction. You found that conference very fruitful. We will work with you all, because our Chancellor used to say, together with you, we will make a difference in this world. Together with you, we will create new innovation. So with this, my best wishes, and thank you so much for inviting me to be here at the valedictory. God bless you.
Thank you, sir, for the insightful address. I now request Professor Dr. Amitabh Mukhopadhyay, co-chairperson of the conference and Professor Amity Institute of Biotechnology, to share his conference impression reflecting on the key highlights and takeaway of this enriching conference.
Good afternoon. Respected Founder President Sir, respected Chancellor Sir, respected Vice Chancellor Madam, and other dignitaries in the podium as well as in the auditorium. First of all, I should not be part of these validation functions. But once I'm called, I need to speak something. The success of a conference depends primarily on three things. The quality of the speaker. I think we have extremely, what do we say? The speakers who are well known in the country and abroad. So these speakers are almost all of several of them are Bhatnagar awardees, and I think we have around 9 directors from the national laboratories. That provides an opportunity to show the Amity University Noida to these national leaders, and most of them are first visitors into this campus. I hope they appreciated our effort, and we had really a brainstorming sessions for last 3 days, which helps us to learn, especially the students must have learned something which will help them to think about and build their future. Second thing, another reason to a successful conference is the money. If you don't have money, you can't do anything, whatever way you want to improve. So I think a large number of sponsors wholeheartedly supported us to organize this conference. So they also deserve a big hand. Thirdly, the success of the meeting depends on how much hard work we can put together. And for me, I always like to work in the background to best of my ability. And I do things in an informal way, which people may or may not like it. So that's the reason I said I don't deserve any credit, or I don't need any. All credit goes to the team AIB with Pooja's leadership to organize this conference successfully. I hope I'll end my talk here, and I will wish all success to the university, especially to AIB. Thank you.
Thank you, sir. Now I request Dr. Edithil Vijayan, President SBTI, to share his valuable experience and insights about the conference.
Good afternoon, ladies and gentlemen. We have been having the annual meetings right across India since 1995, starting from Kerala, Tamil Nadu, Andhra Pradesh, Maharashtra, West Bengal, Uttar Pradesh, and Gujarat. All these years in different. I must confess, and I'm happy to say that by far, this is the best conference of SBTI. That doesn't mean earlier, but we use scientifically, every year we used to have excellent intellectual entertainment. That means the scientific quality was second to none in all the meetings. But this meeting, because of the status of the Amity University in the country, and also the driving force, right? From the founder, the day for opening day, the founder came on the screen, and such an encouraging, stimulating, and inspiring lecture he gave to all of us. And the Vice Chancellor Pro-Vice Chancellor, and the chairman of the department, and so vibrant, active Puja, and Manasri. So what we found was the entire university administration, right from the founder, was behind the organizing committee and sought every aspect in very detail, whether it is arrangement, the logistic, hospitality, food, you just say everything they have their touch. Because of that, we have a very, very successful conference. And scientific presentations, I don't want to repeat, all of you know what we have. It was really of the top order. There's all the top order scientists who are present on the podium and presented the various aspects of the scientific achievements in the country. They're all heading national institutions. So you can imagine what they presented was the summary of what is being done from their respective institutions for the progress of the country. So when you listen to such wonderful presentations, and when you compare what is going around the world, in other part of the world, the scientifically, we are second to none. Not in every aspect of science and technology, India has come to the forefront, and we are on the presence of the youngsters. The youngsters are, as Dr. Selvamurthy mentioned, they are the future, and it is very heartening to see that so many, in hundreds, the youngsters are interested in biotechnology. I don't want to repeat, and he has already mentioned where we stand. That is the future. What biotechnology is different from other branches is that it provides you an opportunity to be an entrepreneur by yourself. You don't have to seek a job from somewhere. You can create job yourself as well as to your colleagues. So that's one interesting aspect of biotechnology. So the great future is in biotechnology, and the society appreciates and thank MIT University for helping and arranging and conducting the annual meeting, along with an international conference with such brilliant scientists presenting their work in various aspects of biotechnology, applied aspect of biotechnology, which is definitely motivating and inspiring the young students for their future. So with these few words, once again, on behalf of the society, I thank MIT University authorities and the organizing committee for organizing such an excellent meeting. And we have one more. The next meeting is going to be another institution of eminence in the country, SRM University, Amaravati. So I told yesterday, Dr. Professor Jayasilan, that you will have to beat what you have seen here. It will be really tough. So you can imagine, because they have set such an example of such a highest order in every aspect. So he will have a tough time to come near to that. So I'm sure, I'm sure he will try to overdo it. So I will, on behalf of the society, I will invite all the students to participate in the next year conference. That will be somewhere between October and December, that is 2026. The dates will be decided later, and we have, you can see in our website, and also we have the newsletter which is published in the website. So with these few words, I once again thank all the organizing committee members and the university for giving us the opportunity to come here and have the meeting. Thank you all.
Thank you, sir, for your kind words. May I now request Professor Dr. Sudeep Bose, co-chairperson of the conference and head Center for Medical Biotechnology, to present the conference report highlighting the scientific sessions, key deliberations, and major outcome of ICoMB 2026. Over to you, sir.
Thank you, Dr. Rajshri. So good afternoon to all eminent speakers, dignitaries, faculties on the dais and off the dais, and to all my dear students who are present in this hall. So it's my great pleasure as the co-chairperson to present the report of the International Conference on Medical Biotechnology, ICoMB 2026, organized by Amity University in collaboration with Society of Biotechnologists India, SBTI. As we reach the end of the ICoMB 2026, I stand here with a heart full of gratitude and minds inspired by the brilliant scientists we have witnessed for the past 3 days. Eminent speakers have delivered a wide spectrum of lectures on the different aspects of the biotechnology, starting with the basic research from cell-to-cell interaction, protein, RNA, DNA interactions, cell immunity of various diseases, cancer to parasitic diseases, and immunotherapy, targeted therapies, and so on. We conceptualized the conference at the AIB to create a platform where bench-to-bedside translation isn't just a concept, but it's a reality. To make this conference successful, it is supported by different funding agencies, Department of Biotechnology, ANRF Anushandan National Research Foundation. We have the industrial support from the Biorad, Zeiss, DSS, Genine, Molecular Devices, Horiba, Merck, and so on. Towards this goal, we began this journey with a pre-conference workshop, and the workshop pillars are from the nanotechnology, flow cytometry, FACS, colonoscopy, clinical diagnostics, confocal microscopy, and advanced imaging. On the 6th January, we did this workshop with the technical sessions, 8 technical sessions. The research centers involved 4, and experts and mentors and speakers have more than 10, and more than 65 participations. Total number of sessions conducted for this conference were 14 sessions. Total number of speakers, more than eminent 60+ eminent speakers were there, and more than 350 students around all over parts of the India. The session started with the inaugural ceremony on 7th of January with a welcome address by our AIB Director, Professor Pooja Vijay Raghavan, opening remarks by Additional Pro VC and Dean Bioscience and Biotechnology, Professor Dr. Chandradeep Tandon, addressed by Professor Dr. Edithil Vijayan, President of SBTI, and special address by Professor Dr. Balvinder Shukla, our Vice Chancellor, ma'am, inaugural address by INSA President Dr. Shekhar Mande, and Lifetime Achievement Award recipient Dr. Madhu Dikshit, followed by release of conference souvenir and blessings from our Honorable President, Dr. Ashok Ken Chauhan, sir. The conference themes, there are 10 themes are there, from molecular mechanism of cellular regulation, cancer biology and cell signaling, stem cell research and regenerative medicine, microbial pathogenesis and infection biology, immunotherapy and immune technology. neuroscience, brain health and neuro-oncology, biomaterial sciences, nanomedicine, multi-omics and therapeutic targeting, translation, biosciences, and public health. The eminent speakers from different parts of India are Dr. Shekhar Mande, Bhatnagar Avadi, Dr. Deepak T. Nair, Dr. Pushkar Sharma, Dr. Shuman Dhar, Dr. Rana Pratap Singh, Vice Chancellor, and many others. The international speakers are Dr. Darren Tomlinson, Dr. Thomas Hughes, Dr. Efrain Rice and Donna Jones, Dr. Tushar Komeria, Dr. D.M. Siddiqui, Dr. Momita Datta, and many others. So our industrial participation, which also there they delivered the lectures by Ms. Shobha Iyer, Mr. Akhil Gupta, Mr. H.C. Sudeksha, Mr. Ranjit Chaudhary, Amit Bhatt, Hemant Kavalkar, and Sridhar Tiwari. To bridge the gap between the basic science and the medical thing, we have a panel of clinical perspectives where various doctors have also delivered their lecture. Dr. Bikram Datta, Dr. Rohit Kumar, Dr. Rohan Sinha, Dr. Vedant Kabra, Dr. Kunal Das. From SBTI has supported with the different awards categories which have been given to the students and the scientists. Padma Bhushan, Professor M.V. Piley Lifetime Achievement Award, Dr. C.S. also Memorial Oration Award, Young Scientist Award, KPL Award, IBS Award, C.V. Jacob Award, and many others. So, at the last, I would like to give, without their support and blessings, this conference was not successful. I'm really heartfelt thanks to our founder president, sir, Dr. Ashok K. Chauhan, our chancellor, sir, Dr. Atul K. Chauhan, Abhay Chauhan ji, Amol Chauhan ji, our beloved vice chancellor, ma'am, Dr. Balwinder Shukla, our Chandadeep Tandon, sir, our ex-director, and Dr. Pujay Vijay Raghavan, our director of AIB. She belongs to my center. That's my privilege. I'm very proud of that. And the main instrumental part of this conference is Sir Dr. Amitava Mukhopadhyay and organizing secretary, Dr. Banashri Bandopadhyay. I would like to thank all the faculties of my center who work day and night for past 2-3 months for different teams, different organizing teams. And I'm also thankful to different other institutes, sister institutes, Professor Subrajit Biswas, Dr. Manoj Garg, Dr. Amit Kharakwal, Dr. O.P. Sinha for helping them in the, Dr. Kumud Bala for making this workshop and this conference successful. And not last, the all student volunteers whose dedication, enthusiasm, and teamwork ensured the smooth and successful execution of this conference. With this, I would like to share glimpses of our 3 days conference in a videography. Sounded. you so much.
Thank you, sir, for that comprehensive overview. I now invite Professor Dr. Chandradeep Tandon, Additional Pro Vice Chancellor and Dean, Faculty of Biosciences and Biotechnology, to deliver his address and share his insights with us.
Thank you so much. Most revered Founder President, Dr. Shokesh Chauhan ji, Honorable Chancellor, Dr. Atul Chauhan ji, Honorable Vice Chancellor, Dr. Vrinda Shukla Madam, who is here with us throughout this conference, and all dignitaries on the dais, off the dais, my esteemed colleagues, students. As we come to the conclusion of ICoMB 2026, it gives me great satisfaction to reflect on the rich scientific deliberations and the vibrant exchanges that have taken place over the past days. You know, whenever I'm in front of all my colleagues of AIB, I get very nostalgic. I remember when I joined in 2014 as the Director of Biotechnology, the responsibility given to me by Honorable Founder President Sir, Chancellor Sir, and Vice Chancellor Madam. Founder President Sir told me that you are joining an institute where the faculty members are the best faculty members. And the, per square area, if you look at the intellectual capacity, he told me that is the best. And believe you me, I am telling you, it is not only the best, it was amazing. My 7 years as a director of AIB, I could see the potential in all my colleagues, the way they performed, and whatever, when it comes to research or academics, funded projects, patents, publications. So whatever targets were given to them, they tried their best to achieve it. I'm so proud of our AIB. And not only AIB, when I became Dean, Vice Chancellor Madam told me, it's not only AIB, the whole domain, which you are going to be, for which you will be responsible as a Dean, is one of the best domains. And Madam, actually it is the best domain. I mean that I must salute this. All our HOIs and all the faculty members, all colleagues, even the non-teaching staff, they are very good. And we expect that not only because of this conference, otherwise also they should keep on performing. Now coming back to the conference. The scientific discussions which took place, they seamlessly traversed molecular insights, disease mechanisms, translation research, clinical relevance. Cross-country and cross-institutional interactions enabled the exchange of best practices, comparative research perspectives. Industry-focused sessions, I'm going to be very brief. Industry-focused sessions brought valuable insights into scalability and regulation and commercialization of diagnostics. Now, having said that, we have had, we have various conferences, but the USP of this conferences is, actually, there's not a single USP of this conference. I'm changing the connotation of the word USP. There were many USPs of this particular conference. The first one, by the combined team effort of led by Dr. Pooja under the mentorship of our Vice Chancellor Madam, Amitabha ji played a pivotal role. I know he's a very humble fellow. He always says that it is not me, I don't need a credit, that is fine. But a fact remains fact. It is his good networking by virtue of which we had a galaxy of directors from all the topmost premier institutes, which is something which is unique, which has happened. And this provides, I'm telling you, the positive effect of it is going to go a very long way. MIT University has already carved a niche for itself in the whole world, in India and across the whole world. But yesterday's interaction, when we had a special session with all the directors, that was something unique, where Vice Chancellor Madam felicitated each and every director who came there. And now they very well know whatever MIT University used to say that we are the best, we actually are the best. So we must put our hands together for all of you, for that. The SBTI awards and recognition presented in the conferences celebrated lifetime contribution to the biotechnology, excellence among young scientists, and outstanding research presentations. Dr. Vijayan, who is with us today, I was so happy when he, expressed that it is perhaps one of the best. He was, very generous in his expression. He said it's perhaps the best. So we really respect that, sir, for that. And we promise you that we'll continue to keep this bar. We'll keep on increasing the bar high and high till we touch the sky with the blessings of God and our wonderful President, sir. So extend my sincere gratitude to all speakers, delegates, organizers, sponsors, and partners for making this conference a great success. So with great optimism for continued collaboration and scientific progress, let's put our hands together and thank you so much. Thank you.
Thank you, sir, for your inspiring words. We are deeply honored to have Professor Dr. Balwinder Shukla, Madam, Honorable Vice Chancellor, Amity University, Uttar Pradesh, with us today. A visionary academic leader, Professor Dr. Balwinder Shukla, Madam, has been instrumental in steering Amity University towards global excellence through innovation-driven research, international collaborations, and a holistic approach of education. I welcome ma'am to deliver her special address.
Thank you so very much. I cannot really express my happiness. I'm too happy and delighted to be here for the conference and what I have witnessed in last 3 days. What our Honorable Founder President says that bring the word to Amity and take the Amity to the world. This Amity being the research and innovation-driven universities. The 3 days, this campus have been breathing, talking, and living really the scientific environment. And this is what I was expecting from AIB for quite some time. I think 2026 have been really the year where we have very first week itself, we have started this very important conference. And I think now that AIB has geared up to have the series of conferences. I'm also happy because this year is the AIB's the Silver Jubilee year also. Completion of 25 years. You know that, look at the vision of Honorable Founder President when AIB was initiated. And we had first the Sector 44 and then this campus and A block for AIB, and then the engineering came and then the other institution came. And today that which was the small seed and became such a big banyan tree, AIB, and known across the globe. And this momentum, why I myself and Dr. Tandon got late for this valedictory session. Honorable Founder President was speaking, and what Dr. Lamoti shared that founder also sent the email to Dr. Pooja with copy to us. Because important is not just you do a conference and forget about it. Because the conference is for the student, for learning, for enhancing the knowledge. But it's also a great opportunity for the faculty. What the mission of our Honorable Founder has given, the Mission Connect. The Mission Connect is fulfilled. But then the next mission is a mission synergy. And our Honorable Chancellor always says, build the synergies, whatever the connect you make. And that synergy should be so powerful that which can change the world. And those synergy can be powerful only when you collaborate, cooperate, and which are mutually benefit. If it is a one-sided benefit, relationship will never work. Maybe one time, but not forever. And we always look for long-term relationships. This AIB, when we are talking about, we have a number of faculty who are right from the beginning even till today. We have the alumni of the very first batch. So the MC, which we have, one of them, the Shajia. Shajia, our alumni of very first batch, part of Amity family. Dr. Rachna sitting there, the member of the faculty from the founding years. And that's what is the strength of AIB. The alumni, the faculty who are all building. And our, from founding director to the current director, who all have led the institution so passionately, building the strong teams. And I can see the Center for Medical Biotechnology, which is hosting this, organizing this conference, and next is the Plant Biotechnology. Their conference is coming, and then is the Bioinformatics Center, and all are now geared up. And I think the year 2026 is going to be the year for biotechnology institution of conferences in all domains to build a strong connect with the industry, to build the strong connect with the scientists, researchers, and the professional bodies like SBTi. I'm extremely thankful to the president SBTi that who has, agreed to be the partner with the Amity Institute of Biotechnology to host this conference. And I'm thankful that the feedback which we have got from here, that the conference was the best conference. And it has motivated us that we need to do even better than this. Because for ultimate, it's you can only talk about the journey, but it can never be a destination. Excellence has to be all the time working, and I'm now for Rachna and her team, they have to work very hard because this is the conference, is the benchmark for them, how they do the better one. And so Dr. Nan, as a dean, Dr. Nan, as a dean, you have very big responsibility. As you will be working from today itself, what is the outcome of this conference? You cannot measure the outcome in one day because something you achieve immediately, something you achieve over time, till you feel that we have, whatever we envisioned, we have achieved everything. But in process, you will build very strong long-term relationship associations, which will go for long. I remember there's some of the company who got associated with us way back in 1995, 96. They are associated with us today also. They start associated with us for management domain, but today they are for the biotechnology, they are for engineering and technology, they are even for law. Because as the organizations grow, so the relationships also becomes the manifolds. And that's what we are looking at. And and also that the students, especially for you people, is extremely important. So what you have learned today, yesterday and day before yesterday in various sessions, from that you have to make your own career path. These are the so many opportunities are there, so many developments are happening. What should I choose for myself? And how should I make my career that I contribute to the Viksit Bharat? Because look at that, what The dream is the India to be the Viksit Bharat by 2047. And you will be at the peak of your career at that time. And you would have contributed a lot. You have to start thinking, dreaming, planning what all you will do. And it is this nation, that's what our Honorable Chancellor always says, and he believes. Youth is the biggest power. And this youth, whether they are within the country or outside country, they will contribute enormously in the growth of the nation and also make this world a better place to live by your research, by your innovation, by your ideas, and what you contribute. With this, once again, I would like to thank all the distinguished speakers who have come and presented their thoughts, their words of wisdom, and also all the sponsors, whether from the industry or from academia or from the government, who supported this conference. I'm sure that we have met their expectations. And also I would like to thank the organizing team under the leadership of our Dean, Dr. Tandon, supported by Dr. V. Pooja, the head of the institution. She is the leader. She have, but guidance, support of Dr. Tandon have been immense. And the biggest thanks is to Dr. Mukhopadhyay, because he has leveraged on his contacts, brought them to the MIT. But the synergy of energy of all the faculty of MIT Institute Center of Medical Biotechnology. So big thanks and congratulations to the team. And I'm sure the other centers faculty have learned a lot, and they will outshine very soon the Center of Medical Biotechnology. So thank you so very much, and congratulations, and especially my student volunteers, because you are the energy of the university. You are the brand ambassador of this. You are the product. How the product of MIT performs, that builds the reputation of the universities. So therefore, my best wishes to all the students, because many of you are the final year and you will be looking for your progression. And that, as Dr. Pooja has shared with me, the 60% of the students have already got their placement, just 40% are left. So I wish all the best to all the students, that all of you have your career aspiration fulfilled. Wish you all the best. Once again, I would like to thank our Honorable Chancellor, Honorable Founder President, for their guidance, for their blessings, that for making this conference a great success. Thank you so very much.
Thank you, ma'am, for always motivating and guiding us, and for unwavering leadership and vision. We now move on to one of the much awaited prize distribution ceremony of the valedictory session. May I now request Professor Dr. Balvinder Shukla, Madam, Professor Chandradeep Tandon, Dr. Erithil Vijayan, President SBTI, to kindly present the SBTI awards. I also request Dr. Nigel George, Treasurer SBTI, to join the presentations award ceremony for presenting the awards. I request Dr. Anjutia, Secretary SBTI, to announce the name of the award winners.
Respected dignitaries, colleagues, scholars, students, ladies and gentlemen, good afternoon. The most inviting moment is here, and I'm very happy to declare the SBTI Young Researcher Award. First of all, let me congratulate all the presenters. All the presentations were wonderful, but we have awards for only 6 best presentations. And here goes the awards. While I announce the awards, I request the awardees to come forward and
receive the awards. First, Kunnat Pharmaceutical Award for Herbal Medicine goes to Shinjini Sen from MIT University. Congratulations. Narasimhaya Award for Cancer Biology goes to Kasturi Ganguly from National Institute of Immunology, New Delhi. Congratulations. Praise C.B. Jacob Award for Plant Biotechnology goes to Ritu Koshla, Acharya Narendra Dev College, New Delhi. Congratulations. IBS Award for Medical Biotechnology goes to Abhinash Datta, NIST University, Odisha. Congratulations. KPL Award for Industrial Biotechnology goes to Tanmay Jayus, Amity University. Congratulations. Professor Idathil Vijayan Award goes to Nailanjan Maitra, Amity University. Congratulations.
We now proceed with the Oral Presentation Awards, recognizing excellence in scientific content, originality, and presentation skills. The first position goes to Rupa Mishra Amity University, Uttar Pradesh. Second position goes to Dr. Manish Kumar, AIMS, Jammu. I think we don't have Dr. Manish here. We have another second award, which goes to Agnish Pratim Das, ICMR, NICPR. We have appreciation awards. The first goes to Jyoti Kiran Bara, Barkatullah University, Bhopal. Yes, sorry, appreciation certificates. On second place, we have Sarthak Dahiya, AIB, Amity University. Okay. The third place goes to Sanchita Roy Pradhan, AIMMSCR, Amity University, Uttar Pradesh.
We now move to the poster presentation award, acknowledging innovation, clarity, and effective visual communication of research. For this, the first prize goes to Harshada L. Lokhande, D.Y. Patil University. A big round of applause, please. The second prize goes to Muskan Syed, Amity Institute of Biotechnology, Amity University, Uttar Pradesh. The third prize goes to API Talom, Rajiv Gandhi University, Arunachal Pradesh. We have two appreciation certificates. First, it goes to Ria Sahani, University of Lucknow. Miss Ria Sahani. Malika Ranjan, AMSR UUP.
The second prize for oral presentation, Mr. Agnish Pratim Das, ICMR NICPR. He is here. Please come on the stage to take your award.
May I now request the dignitaries on the dais to kindly felicitate the jury members as a token of sincere gratitude for judging the poster and presentations. I respectfully request the jury members as I call out their names one by one. The judges for oral presentation, Dr. Neil George. Pardon me, Dr. Nigel George.
Dr. Nigel George.
Next is Dr. Anju TR. Dr. Jayasila Murugian. Dr. Anil Suresh.
Okay, sir. Dr. Anil Suresh is not here. So judges for poster presentation, Dr. Ashish Gupta, Dr. Bishuru Basu, Dr. Bishuru Basu is not here. Professor Deepak Ganjewala, Dr. Devinder Toor, Professor Shoma Paul Nandi, Dr. Sonia Kapoor, Dr. Yogita Adlaka. Thank you. That brings us to the end of the prize distribution ceremony. As we approach the conclusion of this valedictory session, I now invite Dr. Banishri Bandyopadhyay, Organizing Secretary.
The organizing team will like to felicitate the special people without whose support it was impossible to hold this conference. They are the strength and instrumental in mentoring, guidance, and shaping the conference. The first person is Honorable Vice Chancellor, Madam Dr. Balvinder Shukla. We will also like to felicitate Professor Dr. Chandradeep Tandon, without whose support this would not have been possible. After the vote of thanks. As we approach the conclusion of this valedictory session, I now invite Dr. Banashri Bandopadhyay, Organizing Secretary of ICoMB 2026, to deliver the vote of thanks.
Respected distinguished guests, dear colleagues, and my dear students, as the organizing secretary of the International Conference on Medical Biotechnology 2026, that is ICoMB 2026, at Amity University, Uttar Pradesh, Noida, it is my immense honor and deepest gratitude to present this valedictory vote of thanks on behalf of the entire organizing committee. Over the past 3 days, ICoMB 2026 has been a resounding success, bringing together global minds in medical biotechnology for inspiring discussions, insightful presentations, and meaningful collaborations. I would hereby like to thank each and every person who has been involved, be it our distinguished guests, organizing committee members, or our beloved student volunteers. Your collective efforts and selfless commitment have elevated this conference to its highest success. I would first like to extend my heartfelt gratitude to our Honorable Founder President, Sir, for his constant blessings, which have served as the guiding light behind every success at Amity University. I also extend my sincere gratitude to Honorable Chancellor, Sir, for his exemplary leadership, which has consistently inspired us to achieve the highest standards at Amity. We, of course, express our deepest gratitude for the unwavering and visionary support at all times towards our honorable and beloved Vice Chancellor, Madam. We are proudly grateful to Dr. W. Selva Murthy, Sir, President, Amity Science, Technology and Innovation Foundation, ASTIF, for his distinguished patronage and continued support in making ICoMB 2026 a landmark event. We also extend our appreciation to our beloved and honorable respected Professor Dr. Chandradeep Tandon, Sir, Additional Pro Vice Chancellor and Dean, Faculty of Biosciences and Biotechnology, for his constant guidance during the conference planning and execution. We extend our sincere gratitude to Professor Dr. Idathil Vijayan, President of the Society of Biotechnologists, India, for your esteemed presence and invaluable contributions to ICoMB 2026. Your thoughtful guidance and deep passion for the field have inspired everyone gathered here. We are grateful to the SBTI members, including Dr. Anju TR, Secretary SBTI, and Dr. Nigel George, Treasurer SBTI, for choosing this platform for hosting the annual body meeting. We are honored to have received support from DBT, ANRF, and CSR, along with our industry partners, RAR Life Sciences, DHSys, ImageTech, Merck, Horiba, Leica Microsystems, Molecular Devices, Carl Zeiss Private Limited, Genai Laboratories, New England Biosystems, MacFlow Engineering, PK Scientific, EduDAP, and Laxine. We extend our heartfelt appreciation to all the esteemed speakers and session chairs, juries, as well as the co-chairs of ICoMB 2026 for your insightful presentations and thoughtful moderations. Your expertise from leading institutions across India and abroad has enriched every session, sparking innovation and collaboration that will shape the future of medical biotechnology. Thank you sincerely for your time and support. We extend our deepest gratitude and heartfelt appreciation to our beloved and respected Professor Dr. Pooja Vijayaraghavan ma'am, Director of Amity Institute of Biotechnology and Chairperson of ICoMB 2026, for your exemplary leadership, meticulous guidance, and unwavering commitment throughout this conference. Thank you profoundly, ma'am. Our sincere appreciation goes to Professor Dr. Amitabha Mukhopadhyay, sir, the esteemed co-chairperson of ICoMB 2026, for your visionary leadership, strategic guidance, and unwavering commitment throughout this conference. Your profound expertise in steering complex initiatives have been instrumental in shaping this conference. We extend our deepest gratitude to Professor Dr. Sudeep Bose, sir, the esteemed co-chairperson, for your insightful oversight, dedication to the scientific excellence, and skill facilitation. Our heartfelt thanks goes to dedicated organizing committee of ICoMB 2026, whose coordinated effort and unwavering commitment made this event a success, a grand success. Thank you sincerely to all my beloved and respected colleagues of the Center for Medical Biotechnology. I'll quickly take everybody's name. I've already taken that permission. Dr. Girish Sharma sir, Dr. Kumud Bala ma'am, Dr. Vishwajit Shah sir, Dr. Rachna Sahane ma'am, Dr. Rashri Das ma'am, Dr. Amitabha Mukhopadhyay sir, Dr. Sarika Saxena ma'am, Dr. Nidhi Srivastava ma'am, Dr. Madhumita P. Ghosh ma'am, Dr. Navkiran Kaur ma'am, Dr. Gayatri Sharma ma'am, Dr. Nahid Siddiqui ma'am, Dr. Devlina Ray ma'am, Dr. Vijay Kumar sir, Dr. Rajpal Srivastava sir, Dr. Surbhi Sinha ma'am, Dr. Priyanka Chabra ma'am, Dr. Arindam Pramanik sir, myself also, Dr. Shruti Khanna Ahuja ma'am, Dr. Chajia Rashid ma'am, Dr. Sushil Kumar Bhushan sir, Dr. Abhijit Ghosh. I hope I haven't forgotten anyone's name. And to all our participants, we are deeply grateful for your engaging participations. We truly hope that this conference has opened up many new opportunities for you and has given you inspiration to achieve the best. In these 3 days, we had multiple poster, oral, and Young Scientist Award presentations wherein participants have eagerly presented their work and gained experience. On that note, I would once again like to wholeheartedly welcome you all and say a thank you to this international conference and to many more upcoming conferences at Amity University, Uttar Pradesh, Noida. Last but not the least, our heartfelt thanks goes to the dedicated student volunteers. 50 of our student volunteers had worked relentlessly for this conference success. This is a big round of applause for you guys. You made this conference a big success. Thank you to all my student volunteers. Whenever I call them, either at early morning, 5 o'clock, or at night, 1:45 a.m., everybody on a single call has been there to assist our speakers, participants, everybody. All the speakers, participants, whosoever come here, has graciously told me that you had amazing support from your students. They did wonders. Really, I'm so thankful to you guys. And at last but not the least, and like Nelson Mandela has famously said, it always seems impossible until it's done. And with that note, we bid a goodbye to ICoMB 2026.
Before we bid goodbye to ICoMB 2026, I would take this moment to thank our media team, the entire team of admin of Amity University, our team which supported us immensely, Wing Commander Sharma's team, every single person from Amity University, Dr. Chaya, the hostel team, the guest house, everyone who supported us and ensured that we do not, get anxious. So thank you, everybody. And on my own behalf, I would like to thank each and every faculty member. The stage is small. We are so many. This is the strength that we have. We need to ensure that we harness the strength to its fullest limit. Thank you so much, each and every person of AIB who came forward to support. And I would request Dr. Banashri Bandyopadhyay to announce the next year's conference as well.
So on next year also, again with the same name, we'll be coming back as ICoMB 2027. And we hope to get the same love, support, and cooperation from you all. Thank you.